Hello my dear friends, welcome to Live Prep. Myself Dr. Gajapati Verma. As I informed you earlier, under the name of Bootcamp, we are providing both revision lectures and also we are conducting test series. Revision lectures are available both on Live Prep app and also on our YouTube channel. But the test series can be given only through the app. Now, in this lecture, we are going through the revision of the chapter Neural Control and Coordination. And here please remember, and in this latest edition of NCRT, they have reduced this particular chapter. And please remember, we don't need to go anything out of NCRT. If you can understand the information given in NCRT, that is more than enough. Because in this chapter, if you want to study something extra, so there is no limit for that extra. So unnecessarily you will end up in something that is not related to NCRT. So that's why my suggestion is please follow NCRT only. Clear? And now we are going with this particular one. And here one more point I wanted to tell you is, since the information related to that nervous system, if you specifically see, so regarding the spinal cord, they have not given any information. Regarding the cranial nerves and spinal nerves, they have not given any information. So leave it. And sensory organs, they have not given anything. So these informations are not needed at all. Clear? And with respect to the brain specifically, simply they have given about the forebrain, midbrain, hindbrain and some names and the functions they have given, and that much is enough. So since that information is very less, so mainly we are focusing on neuron structure, and anyhow neuron structure is also very simple information, you can easily remember. But the major point, and many students are having doubt regarding nerve impulse generation and conduction. Understood or not? So that's why I will also give more emphasis on that particular one, and that too in the NCRT, that nerve impulse generation and conduction information was not clearly given. So that's why what I will do is I will explain you some concepts and that are related with again this particular process, and at the end I will try to give some simultaneous diagrams and I will try to interlink all these points, and at the same time we will understand many things. So that's why please pay your attention so that you can easily understand this. So, and many students, they are trying to by heart this, so don't do that. Try to understand the concept so that you can easily remember these particular things. Clear? Now we will quickly start with the chapter, and here they are first trying to tell us what is actually this coordination and how this nervous system or the neural system is controlling particular things and how it is coordinating with various other organs. So in this way they are trying to give some introduction. So first we will see here what actually this coordination is. Coordination is the process through which two or more organs interact and complement the functions of one another. So in this way, what is this coordination actually? So nervous system or the neural system is coordinating the functions of various organs. Every organ or organ system is having its own particular function. Okay, they are doing their own functions, but when they have to do, how they have to interlink with each other. So for all these things, someone has to do this coordination, and it is the neural system. And please remember, it is not just the neural system. There is another one that is called as endocrine system. Neural system and the endocrine system, these two are the ones that are doing the coordinations and integrations. Okay, so that's why here in this chapter we are mainly focusing on the neural system. Clear? And here what is the main aim of coordination? And this coordination is to maintain the homeostasis so that an organism can perform various functions and also the organism is able to show some behaviors. Yes or no? For all these things, coordination of these organs is very important. That's why specifically for the coordination there are some systems like neural system and endocrine system. Clear? In this chapter we are going with the neural system. Next one. In our body, neural system and endocrine system jointly coordinate and integrate all the activities of the organs so that they function in a synchronized fashion, so that they are helping in normal functioning of the individual. That's what they are talking. And then we are going with the neural system. In this there are different types of cells are present. But in the NCRT, they have mentioned only about the neurons. In the NCRT, they have mentioned only about the neurons. But not just the neurons, there are many other types of cells are present. Then, what all these cells are together called as? All these cells, except the neurons, remaining all other cells together For our understanding purpose, they are giving a term called as a neuroglia. Neuroglia, please understand this word, neuroglia. So neurons are one type and remaining all cells together we are calling them as neuroglia. Clear? And this is what we are saying. So different cells are there, and what are the important cells, you please understand here. Anyhow, neurons, these are important. Here also we are studying about the neurons. Please understand this. This we are studying under peripheral nervous system. This we are studying under central nervous system. So here we are talking about neurons. This is very important. Neuroglia, not important in this particular class 11th and 12th. Leave it. Astrocytes also not much important. Ependymal cells also not much important. But oligodendrocytes are very important because we are talking about the myelin sheath formation in the central nervous system around the axons of neurons. Who will form the myelin sheath? It is the oligodendrocytes. Please remember this. Okay. Now come to the peripheral nervous system. Here neurons very important. This satellite we are not studying, so leave it. But Schwann cells, yes, very important. And here very important is that in the peripheral nervous system around the axons of neurons, who will form the myelin sheath? It is the Schwann cells. In the NCERT diagram, so around the axon of the neuron, they have mentioned the Schwann cell. So you know that Schwann cells can form the myelin sheath around the axons of neuron. This information you know based on NCERT. But here the important point is, in the case of central nervous system, who will form the myelin sheath? It is the oligodendrocytes. It is not the Schwann cell. Please understand this. Very important one that you need to understand. Next one, neuroglia make up more than one half of the volume of neural tissue in our body. So neuroglia, what are these neuroglia? Other than neurons, all the cells combinedly they are called as neuroglia. And these neuroglia will make up more than one half. More than one half means what? Suppose you are considering completely all the cells together as 100%. One half will be what? 50%. Other half will be 50%. So more than one half means more than 50%. That is what the meaning is. So more than 50% will constitute neuroglia. That means what is the percentage with respect to the neurons? With respect to only neurons, they are less than 50%. This is what the meaning of this particular sentence, and this was not given in this particular chapter. It was related to some other chapter, but that was deleted. But this sentence is important, so please remember. Now next one, with respect to specifically neurons we are focusing, and what they are saying here, neurons which can detect, receive and transmit different kinds of stimuli. So what is the function of these neurons? So they are able to detect some stimulus. After detecting they are excited, they are getting excited. So in this way they are receiving that information and then they are transmitting it. Yes or no? So in this way different functions are performed by this neuron. This is what they are saying here. Next one, they are talking about the neural system in different organisms. In the case of invertebrates, that is not highly developed, but in the case of vertebrates, it is highly developed. And here, under the example of invertebrates, they are talking about the hydra. In the case of hydra, it is having network of neurons. This network of neurons is also called as a nerve net. Please remember this word. This word was not given in NCERT, but please remember this nerve net. Technically, we are calling this as a nerve net. It is a network of neurons. That's it. So in this way, very simple type of neural system is present in hydra. Next one, they are saying that in the case of insects, this neural system is a bit developed when compared with hydra. And in the case of insect, and this is the example with respect to the cockroach. Cockroach is an insect. So anyhow, this information is related with the cockroach. So you can remember this. In the case of cockroach, we are representing supraesophageal ganglion as brain. Yes or no? Supraesophageal ganglion as brain. That means there is a brain here. And even though brain is present, its role is very limited. And there are many ganglia in different segments. And these are also playing very significant role. So that's why in the cockroach they are saying that even though head is removed, this part it can survive for around 1 week, 7 days. This is what generally they are saying, right? But technically if you are talking, there is a brain. But this is not much developed like vertebrates. It is very smaller thing here. So this is what they are saying here. And that here you can see double ventral nerve cord. Anyhow, all these points, so in the animal kingdom chapter we have studied, so leave it. Next one, now let us focus on the vertebrates. So in the case of vertebrates, this neural system is highly developed. That too, if you are talking with respect to the human, now it is highly developed and very complicated. Clear? And now here they are talking about the classification of this neural system. They are classifying this into central neural system and peripheral neural system. So what are the components of the central neural system? Brain and spinal cord are considered as central neural system. And what is the function of the central neural system? So its function is to process the information and control the various activities of the body. This is what the function of this central neural system. And from this central neural system, that means from the brain and spinal cord, many nerves will arise, and they are going to various parts of the body. So the nerves that are coming from the brain are called as cranial nerves. They are 12 pairs in number. And the nerves that are coming from the spinal cord are called as spinal nerves. Please observe carefully. Cranial nerves are 12 pairs and spinal nerves are 31 pairs. So observe carefully. Brain and spinal cord are part of central nervous system, but the nerves that are arising from these are part of peripheral nervous system. That's it. And regarding this number of pairs, it is not needed for examination. Simply for the information purpose I give. That's it. Next one. And among these nerves, again there are different types of neurons are present. And these neurons, based on their function, generally we are classifying into two types. So that's why in the peripheral nervous system, what are present? Efferent fibers are present, efferent fibers are present. And what is the function of these efferent fibers? From the various organs, they are receiving information and they are sending it to the central nervous system. What is the function of efferent fibers? They are receiving information and giving to central nervous system. And then what is the function of these efferent fibers? From the central nervous system, they are giving information to various responding organs. Understood or not? From the organ to the central nervous system, it is taking information towards central nervous system, that is efferent fiber. From the central nervous system to the particular responding organ, it is carrying information. Now this is called as efferent fiber. So in this way, in the peripheral nervous system, efferent and efferent fibers are present. Clear? And now this peripheral nervous system, again based on various factors, we are classifying into somatic neural system and autonomic neural system. Autonomic means what? Now this neural system is going to the involuntary organs. Some things are not under the control of human will, and now they all will come under involuntary. Now this neural system is sending these neurons and receiving neurons from those particular organs. That's why it is autonomic. Whereas the neurons that are connected with the skeletal muscles, now they will come under somatic neural system. This is what they are saying. So under this autonomic neural system, again based on the function of these neurons, we are classifying them into sympathetic neural system and parasympathetic neural system. This is what they are talking. And within this autonomic neural system, again we are considering another part that is called as a visceral neural system. So please remember, visceral neural system is also part of autonomic nervous system, because visceral organs and their functioning, it is not under the control of human will. So that's why this also will come under autonomic nervous system. But here, what is this visceral neural system? The neurons that are going to the visceral organs, and then the neurons that are coming from the visceral organs to the nervous system. So all these things will comprise this particular visceral neural system. Anyhow, very clearly they have given the explanation in NCERT. Simply read, you can understand this. Simply I am trying to give you the summary of all these things. That's it. Clear. Then we are going with the structure of this particular neuron. First let us complete this basic information, then slowly we will go with remaining other points. Clear? And for sorry. So here we are talking about the structure of the neuron. And since we are talking about the neuron, this is the single cell, right? So neuron is considered as the functional unit of this particular neural system. Yes or no? Functional unit. And here I am trying to give you some information. Before directly going with the structure, we are studying some information, and this information I will compare between the muscle and nerve. Muscle and the nerve. So please observe this very carefully here. So already I told you that we are talking about total 4 types of tissues. What are these 4 types of tissues? First one is epithelial tissues. Second one is connective tissue. Third one is muscular tissue. And the fourth one is neural tissue. Yes or no? In the muscular tissue, we are talking about the muscle. And in the neural tissue, we are talking about the nerve. Yes or no? And here for the names wise, if you are observing, you can find some similarity. So since we already studied the information for the muscle we studied the information, and now this is the muscle. Yes or no? First I will tell you the information of muscle, and the similar type of names I will use here. But instead of using the myo, in the myo muscle we are using a term called as a myo, but in the case of nerve we are using the term called as a neuro. Okay, these are the small difference. Now this is the muscle, and this is a muscle tissue, and in this muscle tissue we are studying about muscle bundles. Muscle bundle, it is the muscle bundle. It is also called as a fascicle. It is also called as fascicle. Already we studied about this, right? And inside this muscle bundle we are finding the neurons. We are finding this muscle fiber, right? So we are talking about the muscle. So inside this we are talking about the muscle fiber. Single cell is called a muscle fiber. You can also call this as myocyte or muscle cell. Any of these names you can use. Yes or no? So in this way, this is the muscle, and this is the muscle bundle or fascicle. Inside this is the muscle cell or myocyte or muscle fiber. These are present. This is what we have studied. Same way, here also we are talking this. Now here what we are talking? Here it is the nerve. Please observe carefully. Nerve is a tissue. Okay, it is the nerve, and inside this there are some bundles. And what are these bundles are called as? Nerve bundle. This is the nerve bundle. Here it is the muscle bundle, here it is the nerve bundle. And in both the cases, the term used is the same, that is called fascicle. In both the cases, it is called as fascicle. And inside this, neurons are present. Inside this, neurons are present. Please observe carefully. Neuron is present. Here it is the muscle fiber, but here it is the neuron. In this way, if you are observing the structure wise, so you can find some similarity. Not only this, there is another similarity. What is this? This muscle is surrounded by a fibrous connective tissue. It is called as epimysium. Yes or no? But in the case of nerve, and this nerve is covered with a fibrous connective tissue, it is called as epineurium. Observe carefully. Here you are using a term called as myo. Here neuri. So in this way we are using epimysium. Here epineurium. Now around each bundle, there is a fibrous connective tissue, and this is called as perimysium. Okay. Same way here also there is a fibrous connective tissue around each fascicle, and this is called as perineurium. Names you can compare so that you can easily remember this. And here around each muscle fiber or muscle cell there is a connective tissue layer. It is called as endomysium. And here it is, here also there is a connective tissue layer, and this is called as endoneurium. Understood or not? And here not only this, there are many other cells that are present in the nerve. Okay, so this is what the general information that they are talking, and this information you can compare between these two, and you can easily remember all these names. And this information was not given in NCERT, but better try to remember, because this is a very simple and easy information, and that may be useful for you. And now we are talking about the individual neuron. And what is the structure of the neuron? Very basic thing. So in simple, I try to complete this. And what are the important points? There I will focus more. And in the basic structure of this neuron, we are talking about total 3 regions. What are these? First one is called as cyton or cell body. Second one, these branched structures, they are called as dendrites. And the third one is this long fiber-like structure. This is called as axon. So total 3 regions are there: cyton or cell body, dendrites, and axon. So these are the 3 parts that we are talking. And now inside this axon, cyton, and dendron, this cytoplasm is present. Okay, cytoplasm is present, and the cytoplasm that is present in the neuron is called as neuroplasm. It is called as neuroplasm. But specifically if you are talking about the axon, the cytoplasm that is present in the axon is called as axoplasm. Is it clear? It is called as axoplasm. Okay, nice. Next one. Next one important point is regarding the axolemma. We are talking about axoplasm and axolemma. You need to remember these names because in the nerve impulse generation and conduction, generally these names are used. What is this axoplasm? It is the cytoplasm that is present in the axon portion. That's why axoplasm. That what is this axolemma? The plasma membrane, the part of the plasma membrane for the axon portion is called as axolemma. This is what the name that we are using. And there is a separate term called as a neurolemma. After completion of this, I will talk about the neurolemma. So leave that apart. And this is what the general information is. Like any other cell, it is also having the nucleus, it is also having the cell organelles. So all these are present. But with respect to cell organelles, if you are talking specifically, generally it is considered that in the case of this neuron, centrioles are absent. Please observe this carefully. In the case of neuron, centrioles are absent. That's why generally we say that this neuron is not able to further divide. It can only increase in length, but it cannot divide because it is not having the centrioles. This is one important point, not mentioned in NCERT, but try to remember this. This is one important point. Next point, based on the diagram also you can understand, there are some dot like structures they have mentioned. And what are these dotted like structures? They are the Nissels granules. They are the Nissels granules. What are actually these Nissels granules? They are actually the fragments of rough endoplasmic reticulum. In simple, if you want to say, they are the fragments of rough endoplasmic reticulum. Rough endoplasmic reticulum means it is having ribosomes. Understood or not? So that's why you don't need to say separately that it is having ribosome, because rough endoplasmic reticulum means already it is having ribosome. Then what is the function? Since ribosomes are present, the function of Nissels granules is to produce proteins. Yes or no? The function of Nissels granules is to produce the protein. This is what the function of this particular Nissels granules. This is one important point. Second important point, where actually these Nissels granules are present. Observe the diagram. Nissels granules are present in cell body or cyton. There it is present. Nissels granules are present even in dendrites also. But Nissels granules are not present in the axon. Observe carefully. So Nissels granules are present in cyton or cell body and dendrites, but not in the axon. This is one important point. Clear? And next point is with respect to mitochondria. Mitochondria are present even in the cyton and dendrites also, but mitochondria are more present in the axon portion. Why they are more present in the axon portion? Because, and this axon is a very long portion, and when the nerve impulse is generated and conducted, actually it is conducted in the form of small segments. Segment by segment it is getting transferred. So that's why at that time, so there we will talk about some pumps. For the activity of those pumps, ATP energy is required, and that ATP is provided by mitochondria. So that's why it is having some mitochondria. Clear? This is one important point. Other point we can compare with muscle cell. In the case of muscle cell, we are talking long fiber like myofibrils. Inside the muscle cell, we are talking about myofibrils. Same way, here inside the neuron, we are talking about neurofibrils. Inside the neuron, neurofibrils are present, but we are not talking much information regarding the neurofibrils. Okay, simply remember this. So in this way it is present, and these dendrites they are branched structures. Yes or no? And here this axon, this is the terminal, this is the distal part of the axon. Distal part of the axon is branched, and it is ending up with a bulged portion, and this bulged portion is called as synaptic knob. Inside this synaptic knob, synaptic vesicles are present. Inside the synaptic vesicles, some chemicals called neurotransmitters are present. Clear? And these neurotransmitters, when they are released from this, and then they can transmit the information. How exactly that is taking place? Slowly we will study. Okay, this is about some basic general information regarding this. And finally, if we are talking about the nerve impulse conduction, now actually who will receive the nerve impulse? Dendrites will receive the nerve impulse from the previous neuron, and from the dendrites it will go to the cyton or cell body. From the cell body it is going to the axon. So from the axon it is going to the next neuron. So that's why always the direction of this information transfer or impulse, nerve impulse transfer is from dendrites to cell body to axon. Only in one direction they are going. So this is another important point that we need to understand. Clear? With this part is completed. And finally, now we will talk about the Schwann cell. Regarding the Schwann cell. With respect to Schwann cell, I will draw one rough diagram, so please observe carefully. Now these Schwann cells, actually they are surrounding the axon portion. They are surrounding the axon portion. This is the axon portion. And this is the plasma membrane of the axon portion, right? This is actually completely plasma membrane of the neuron. And here this specifically we are talking for the axon. And the plasma membrane of axon is called as axolemma. Now please observe the difference between axolemma and neurolemma. Many students they are making mistake, and the mistake is, what is neurolemma? They are saying that neurolemma is the plasma membrane of neuron. You should not say like that. You should not say like that. Please observe the difference between neurolemma and axolemma. So plasma membrane of axon part is called as axolemma, correct? Then what is neurolemma? Now observe carefully. Now surrounding this axon, there are some cells called as Schwann cells. This is one Schwann cell. Suppose this is the second one. This is the third one. So in this way they are present. And what is this one? It is the Schwann cell. It is the Schwann cell. And this Schwann cell is having the nucleus, and the location of nucleus is just below the plasma membrane. That means in the Schwann cell, the nucleus is present in peripheral position. That's why it is present just below the plasma membrane. This is what about the Schwann cell. And the plasma membrane of Schwann cell, please observe carefully. The plasma membrane of Schwann cell, not the neuron, it is the plasma membrane of Schwann cell, that is called as neurolemma. Don't make any mistake. So please observe this difference very carefully. Axolemma is the plasma membrane of axon part of neuron, whereas neurolemma is the plasma membrane of Schwann cell. Very, very important point. Many students are making mistake here, so please observe carefully. Next one. What is the function of this particular Schwann cell here? Now please remember very clearly in NCERT they are talking about two types of neurons. Some neurons are called as myelinated neurons. Some are called as non-myelinated neurons. Now observe carefully. Suppose it is a myelinated or non-myelinated, whatever it is, whether it is myelinated or non-myelinated, in peripheral nervous system, Schwann cells are compulsorily present. Understood or not? In peripheral nervous system, around the axon of this neuron, Schwann cells are compulsorily present. But this Schwann cell may or may not produce the myelin sheath. This is important point. Schwann cell is present, but if it is not producing myelin sheath, then the neuron is not having myelin. So that's why now it is called as non-myelinated neuron. So please remember, even though it is a non-myelinated neuron, still the Schwann cells are present, very clearly mentioned in NCERT. So please remember this. So in this way, these Schwann cells are present but not producing myelin sheath. That's why this is example for non-myelinated neuron. Suppose these Schwann cells are producing myelin sheath, where they will produce? On the inner side of the cells. For the cell it is inner side, but exactly if you are talking, it is around the axon. It is around the axon, it is producing this myelin sheath. It is around the axon, it is producing the myelin sheath. This is the myelin sheath. Please observe it very carefully. So this is the myelin sheath. Myelin sheath, what is the appearance of this myelin sheath? This myelin sheath is white in appearance. Myelin sheath is white in appearance. That is the reason why that white matter, gray matter we are talking, no? So why we are getting the white matter? Because that is the myelinated portion of the neuron. Myelinated portion of the neuron will give white appearance. That's why that is called white matter. Whereas the cell body and dendrites, this part, cell body part mainly, the cell body part is grouped together and that will give the gray appearance. Cell body part will give gray appearance, and the myelinated portion of this particular axon, that will give the white appearance. So based on this you are giving white matter, gray matter names. Anyhow, there also once again I will tell you about this. So please remember the logic behind that white matter, gray matter concept. Clear? So in this way, myelin sheath is white in appearance. So that's why it is part of the white matter. Clear? Okay, that point anyhow separately I will tell you. Now here the important point is, if you see the size of the cell, this is the size of the cell. And we are saying that the cell is forming this myelin sheath. Yes, that is correct. But the point is, this myelin sheath is not produced completely in the entire cell part. Okay, only some portion around the axon is covered with this myelin sheath. So this cell is producing this myelin sheath, this cell is producing this myelin sheath. So technically, if you see what you are observing, there is a gap between two myelin sheaths. There is a gap between two myelin sheaths. Observe carefully. And what is this gap between two adjacent myelin sheaths is called as? It is called as, singular is node of Ranvier. Node of Ranvier. Please observe carefully. If you want to say plural, many, if you want to say nodes of Ranvier, you need to say like this. So in this way, the gap between two adjacent myelin sheaths is called as node of Ranvier. This is what technically called as node of Ranvier. Okay, and this myelin sheath is impermeable to movement of the ions. So that's why while we are studying about nerve impulse generation and conduction, we will talk about movement of Na plus K plus all the things. But please remember that those movements do not happen at this myelin sheaths. Those movements can happen only at the nodes of Ranvier. Okay, at those locations only we can observe. So that is the reason why, suppose here that nerve impulse is generated, next nerve impulse will generate at this node, next one will generate at this node, next one will generate at this node. So in this way, nerve impulse generation is happening in a jumping manner. Understood or not? So that's why this type of conduction is called as saltatory conduction. Saltatory conduction. Anyhow, we will study all these things. Saltatory conduction. Is it clear? But if myelin sheath is not there, continuously after one segment, in the next segment, after that next segment, next segment, next segment. So continuously the information will be generated in the normal non-myelinated neuron. That's why in the non-myelinated neuron, the impulse transmission is slow. But in the myelinated neuron, impulse transmission is very quick. Means it is very fast. This is what another important point that we need to link up with the structure of this myelinated neuron with conduction of the nerve impulse. These are all interlinked concepts. You need to interlink them, then only you can understand them very easily. Okay, this is what the basic structure that is related to the myelin sheath. So please remember here itself I am telling you another point. In the case of peripheral nervous system, it is the Schwann cell. It is the Schwann cell. Anyhow, I will show you diagram also. It is the Schwann cell that is important for myelin sheath formation. In the peripheral nervous system, for around the neuron, axons of the neurons, who is forming the myelin sheath? It is the Schwann cell. But in the case of central nervous system, Schwann cell is not forming the myelin sheath. It is the oligodendrocyte. Oligodendrocytes are the cells that are forming the myelin sheath around the axons in the central nervous system. So this point was not given in NCERT, but you need to remember it compulsorily. It is a compulsory and very important information. So this is the basic and general information that is related to the neuron and its structure. Clear? This is what the basic structure related to this. Next we are going with the myelinated and non-myelinated. If myelin sheath is present, they are called as myelinated neurons. If myelin sheath is absent, it is called as non-myelinated neurons. In the case of peripheral nervous system, whether it is a myelinated or non-myelinated, whatever it is, Schwann cell is present. Schwann cell is present. If it produces myelin sheath, that will be a myelinated neuron. Though it is present, if it is not producing myelin sheath, that will be simply non-myelinated neuron. This is what about the myelinated and non-myelinated information given in the NCERT. But if you are technically observing carefully, in the peripheral nervous system, Schwann cells are present. But in the central nervous system, Schwann cells are not present. Then who are present here? Here oligodendrocytes are present. So that's why In the peripheral nervous system, the myelin sheath is produced by Schwann cells. In the central nervous system, myelin sheath is produced by oligodendrocyte cells. So this is the one important point that you need to notice. Clear? Then we are going with the next one. What is the difference between axolemma and neurolemma? Very clearly I told you. The plasma membrane, the part of the plasma membrane present in the axon portion is called as axolemma. It is the axon membrane. The part of the plasma membrane for the axon portion is called as axolemma. Whereas what is neurolemma? The plasma membrane or the sheath of Schwann cell is called as a neurolemma. Don't make any wrong statements in these particular concepts. So please observe very carefully. So this is the Schwann cell that is making the myelin sheath around the axon of the neuron. Okay. So in this up to some portion it is making, and this is also making up to some portion. Between these there are some gap between the myelin sheaths, and that gap between the myelin sheath is called as node of Ranvier. Okay, so technically if you are observing, both the myelin sheaths are present almost very close. Very minute gap is present between both of these Schwann cells. Okay, so anyhow that concept is not needed for you, but technically the what is node of Ranvier? Node of Ranvier is the gap between two adjacent myelin sheaths. Clear? Okay. That's why generally while defining what they will talk, they will not talk with respect to myelin sheath. Generally they will say that the gap present between the two adjacent Schwann cells is called as node of Ranvier. This is what they are saying. But technically if you are speaking, it is the gap between two adjacent myelin sheaths. This is what the better definition. Clear. Next one, type of neurons based on the number of processes. How many processes do they have? So based on this, in the NCERT they are talking about unipolar, bipolar and multipolar. This is what they are talking. What is that polar? What is that polarity? It is not about the polarity, it is about the processes. What are these processes? They are the extensions from the plasma membrane. So that's why now I will draw one diagram here. First one for unipolar, bipolar and multipolar. Now you need to observe what are the similarities and what are the differences. Now observe the diagram carefully. Whether it is unipolar, bipolar, multipolar, whatever it is, it should have the cell body. This is the cell body, also called as cyton. Okay. Unipolar is also having one cell body. Bipolar is also having one cell body. This is the nucleus, let us talk. It is the nucleus. Okay. And here also it is having one cell body. This is the cell body. Please observe carefully. Whatever is the neuron, whether it is unipolar, bipolar, multipolar, whatever it is, compulsorily it is having one cell body. That is compulsory. Now, then what is this uni, bi and multi? Uni means single, bi means two, multi means many. What are these? From this cell body, how many extensions are coming? This is important. From this cell body, if only one process is coming, now it is called unipolar neuron. From this cell body, if two processes are coming, this is one process and this is another process. If two processes are coming, then it is called as bipolar. That means cell body is common for all. But how many processes are coming from this particular one? This all. And here more than two. So this is one and this is another one. And here you can write any number, no problem. So in this, if many processes are present, it is called as multipolar. In this way we are drawing the rough diagram for these particular neurons. Now it is okay. Now here it is only one, and here it is one and one, two are there, here many are there. Now here the next question is, in all these things cell body is there, but in a regular standard diagram of neuron we are studying three parts: cell body, axon and dendron or dendrites we are talking. These are the three things we are studying. But here the problem is, anyhow cell body is there, but here only single process is there. What is this a single process? Is it axon or dendrite? This is very important thing. So please remember, if it is having only one process, definitely it should be axon. If it is having only one process, and that one process should be axon only, it cannot be dendrite. Please understand the concept. Okay, this is what the axon is. Now here what happens, here two processes are there, and please remember always the number of axons is only one. So that's why definitely one will be axon. Definitely one will be axon. If one is axon, the other one will be definitely a dendrite. This will be a dendrite. Okay, dendrite or dendron, anything you can write. Okay, dendrite. Now what about the multipolar? In the multipolar, one is axon, and remaining all, these may be 2, 3, 4, 5, 6, 7, 8, any number it may be, and these will be dendrites. These will be dendrites. I am writing the plural, dendrites. This is what the basic concept is. So in all the types of neurons, cell body is compulsory. If it is unipolar, the single process is axon. Bipolar will have one axon and one dendrite, and multipolar will have one axon and many dendrites. This is how you need to write, and that too not only just the names. In NCERT they gave examples like locations where they are present. Unipolar neurons are generally seen in the embryonic stages. Bipolar neurons are generally seen in the retina of eye, and multipolar neurons are the maximum type of neurons. In NCERT they gave that they are present in the cerebrum. They are present in the cerebrum in simple. So this is how they have given some examples. So please remember these examples. Along with these three things of NCERT, I am adding one more thing. This is also a little bit important because the syllabus is reduced. Now it is not there. But if in the previous one they have given a diagram, it is simply a diagram. So please understand. Now we are talking about another name that is called as a pseudo. Pseudo means what? False. It is the pseudo-unipolar neuron. Pseudo-unipolar neuron. It is like unipolar only, but it is a false. Why it is false? Now we will see. What is unipolar? Unipolar will have one cell body. It will have one cell body. But what is unipolar? It should have one process. If it is having one process, now it is called unipolar. But why they are calling this as pseudo-unipolar? This single process is again branched like this. This single process again branched like two. It is single process from the cell body, so it is unipolar. Though it is unipolar, again it is divided into two parts. So that's why it is called pseudo-unipolar. And in this case, from one side it is able to receive information. From the other side it can transmit the information. From one side it can receive information. From the other side it can transmit the information. Since it is present like this, this type of neuron is called as pseudo-unipolar neuron. And where they are present? Generally these type of neurons we are observing with respect to spinal cord. In the spinal cord generally we are observing this type of neurons. So please observe, in the human also you can see. So in this way these are the various examples of the neurons that we are observing. Clear? With this these names are completed. So very good. But here now I am asking a particular question. So please understand the question first. What is the question? In the case of bipolar and multipolar neurons, dendrites are present. So dendrites will receive the information, and from this dendrite it can go to the cell body. From the cell body it is going to the axon. From this it is going to the next neuron. That is good. That is good. And in the case of pseudo-unipolar neuron, now in this, from this side it is receiving information, from this side it is transmitting information. That is also good. But the question here is for unipolar. In the case of unipolar neurons, dendrites are not present. Then who will receive the information? This is also very important point, right? In NCERT simply they said unipolar, you know the diagram of unipolar, up to that it is okay. But dendrites are not present means who will receive the information? So please observe carefully. If dendrites are not there, cell body can directly receive the information. So this is one important point we need to remember. So that's why please understand who can receive the information. If dendrites are present, dendrites can directly receive information. And if dendrites are not present, cell body can also directly receive the information in the case of unipolar neuron. So cell body is receiving a stimulus and now it is transmitting through the axon. So this is another important point that you need to understand with respect to this NCERT names simply. So this is what about unipolar, bipolar, pseudo-unipolar and then multipolar neurons. These names at least they mentioned. Pseudo-unipolar was not mentioned, but remaining names were mentioned in NCERT. So please observe carefully. Now we are going with generation and conduction of nerve impulse. And in the NCERT they have given such type of diagram, and they are talking about some pumps. They are talking about some channels like sodium channels, potassium channels. And please remember here for this particular concept, I am telling you in advance, I am deliberately spending more time on this because for 90% of the students, they are not able to understand this particular concept. And my aim is, since majority of the portion was deleted, so now in the examination also they will focus more on this particular concept. So that's why every student should understand the concept. It is not just by hearting the things, okay? And in this particular thing we will study different stages like resting stage. It is also called as polarized state. Then we will study about depolarized state. Then we will study about repolarization. Then we will study about hyperpolarization. In this way, total 4 stages we are studying. After hyperpolarization, once again it will come back to resting stage. Now these stages will be repeated. So that's why you need to know about each and every stage of this. And during these stages, there are some things. What happens to during each and every stage? So with respect to sodium potassium pump, is it open or close? With respect to voltage gated Na plus channel, open or close, that you need to know. With respect to voltage gated K plus channel, that also you need to know. And then there are some leaky channels for Na plus and K plus. So these are the 4 things. Understood? 4 stages and 4 of these pumps or channels are there. So now you need to remember all these things. In these four stages, each and everything, which one is open, which one is closed, when it is open, when it is closed. Understood or not? So these are the important concepts that you need to understand. And for that purpose, now I need to explain first about these things. What are these different types of channels and how these channels will work, that you need to understand. Then only you can understand about leaky channels, which is part of this particular nerve impulse generation and conduction. Ligand gated channels, they are not part of nerve impulse and generation, but they are part of this particular diagram. In this diagram, you are actually studying about ligand gated channels. Technically I am speaking. So in this diagram, actually what you are talking about ligand gated channels. So you need to understand this. This is compulsory. Even though don't think that they have not given NCERT, you need to understand. If you want to understand this diagram, means you need to understand about this particular one. Then only you will understand the concept. And voltage gated channels, anyhow it is part of nerve impulse generation and conduction. So that's a voltage gated channel, VGC for Na plus, VGC for K plus, compulsory. And along with this, leaky channels, that is also part of nerve impulse generation and conduction. Along with this, Na plus K plus pump is also important. So that's why if you don't know the individual functions of all these things, and if you don't know when they will open and when they will close, if you don't know these things, you cannot understand that particular nerve impulse generation and conduction concept. So that's why before directly starting that, my aim is first I will explain each and everything, how they will work, when they will be open, when they will be closed. Yes or no? These are the basic concepts that you need to understand. Once you understand these basic concepts, that nerve impulse generation can be understood in just 5 minutes. It will not take 5 minutes. For me to explain, it will take 5 minutes. Understood or not? And once you understood this explanation, for you to recollect this, it will take just 1 minute. And you will never make any mistake in this particular concept. So that's why, please observe these things. So now we need to focus on these things. Please remember, when we study about the ligand gated channels, I will talk about this particular concept. So 50% of this concept you can understand when we are studying about ligand gated channels. Remaining information anyhow separately under this diagram I will explain you, so don't worry. So everything we will cover, okay, but some of the points interlinked are there, so I will repeatedly talking about those things. So please observe these things. Clear? Okay. First, now we are having two things. What is this? Pump is one term. Please understand, pump is one term. Channel, channels. So channels. So in this way, total we are having two names. We are having total two names. First, I'm going with the explanation part of each and everything. Observe carefully. So we are having two things. One is with respect to pump. Other one is with respect to the channels. In general, what are channels? So this information already I have covered under the cell chapter very clearly in the regular lectures I have covered. So if you still have any doubt, you can go through that explanation also. Now here we are talking about active and passive transports. We are talking about active transport and passive transport. Active transport and passive transport. Active transport and passive transport. And these concepts already we studied in the cell chapter. Quickly I am revising this. I will not spend much time on this. What is this active transport? Is energy required for active transport? Yes, energy is required in active transport. In the passive transport, energy is not required. Why it is not required here and why it is required here? Now here the movement with respect to concentration gradient. With respect to concentration gradient, what we need to say? In the case of active transport, the movement of molecules or ions is against concentration gradient. Yes or no? But in the case of passive transport, the movement of molecules or ions is along the concentration gradient. What is the meaning of against concentration gradient? It means that movement of ions or molecules is from low concentration to high concentration. But what is the meaning of along the concentration gradient means? It is from high concentration to low concentration. That's why energy is not required. And since it is the movement from low to high, it is called as uphill process. And here it is from high to low, that's why it is called as downhill process. Yes or no? Already we studied all these names. This is what the general information regarding the active and passive transports. And now here, now we are talking about our content. Our content is based on pump and channel. And actually these are called as membrane proteins. Channels and pumps are membrane proteins. They are present in the plasma membrane. Now the question is membrane protein required for active transport? Yes. For the active transport, always, please remember, if you are talking about active transport, membrane protein is compulsory. It is compulsory. It is required. And with respect to passive transport, if you are talking, two options are there. It may not be required. May not be required. Membrane protein requirement. Membrane protein requirement we are talking. So in the case of active transport, it is required. But in the passive transport, it may not be required. It may be required. Please observe this point very carefully. Why you are writing like may not be required or may be required? Because this passive transport is of different types. Sorry, it is of different types. What are these types? Simple diffusion, facilitated diffusion, and osmosis. These are the three things. Now, where this membrane protein is not required? In the case of simple diffusion. In the case of simple diffusion, membrane protein is not required. These molecules, neutral molecules, hydrophobic molecules, they can simply move across the plasma membrane. They don't require any membrane protein. So in this way here membrane protein is not required. But where the membrane protein is required? Membrane protein is required in facilitated diffusion. It is required in facilitated diffusion. Facilitated diffusion and also in osmosis. Please observe carefully. And in the case of osmosis, there are some membrane proteins required, and these membrane proteins are called as aquaporins. The membrane proteins required for the osmosis process are called as aquaporins. Clear? Very good. Now we need to write about another important point. And now we are coming to the main point. Observe carefully. The membrane protein that is required for the active transport is technically called as a pump. Please observe carefully. This is the very, very important point. Many students are thinking that the term pump is used just normally, casually. No, they are not using casually. Please understand this. This is a technical thing. The term pump refers that it is a membrane protein that is involved in active transport. So that's why if you are talking about a pump means with the help of energy, using energy, it is causing the movement of some molecules or ions from low concentration to high concentration. This is the meaning. So here in this example, in our concept, we are talking about Na plus K plus pump. So what this pump will do? So that we will study. So that's why observe carefully. And here, membrane protein that is required for facilitated diffusion and osmosis. And these are part of passive transport. And here it is called as a channel. Here it is called as a channel. Please understand these things. Many students, they are thinking that randomly these words are used. No, they are having a specific purpose. So if you are talking about a channel, means what a channel will do? Channel is involving in passive transport. That means for the functioning of the channel, energy is not required. And what this channel will do? There are specific channels. If you are talking about sodium channel, it will allow movement of sodium. If you are talking about K plus channel, it will allow the movement of potassium ion. And from where to where? So channel is part of passive transport. So they will allow the movement of these ions from high concentration to low concentration. So these are all the points that you need to keep in mind when you are talking about the channels. Understood or not? But anyhow, we are not talking about the osmosis. Here we are not talking about movement of water. Movement of water is specifically coming under osmosis. So we don't want this. So now we are focusing on the facilitated diffusion of Na plus K plus. Yes or no? So these are the things we are talking. So now in our concept we are talking about the pump, and here the example we are talking is Na plus K plus pump. This is what we are talking. And with respect to the channel, different types of channels. What are these different types of channels? Leaky channels we are talking, ligand gated channels we are talking, and voltage gated channels we are talking. So within the channels also, all these are channels only. They are helping in movement of molecules or ions from high concentration to low concentration only. But how exactly they are working? How exactly they are working? So based on this, there are different types like leaky channels, ligand gated channels, voltage gated channels, and all these three types are related to our concept. Our concept. That's why I'm talking about all these things. So now first I will explain about Na plus K plus pump. Then I will explain about what are leaky channels, what are ligand gated channels, what are voltage gated channels. And this explanation will take a little bit more time, but don't get confused, don't get bored, and these are all very important. Once we complete this, that impulse generation and nerve impulse generation and conduction can be completed in just 5 minutes. Just 5 minutes. You can understand this because these are the logical things. Once you understand the logical part, main information you can understand in just 2 minutes. Okay. Now we will go one by one. First we will focus on Na plus K plus pump. Now please observe carefully. First I am talking about a cell. And here in this case, though I am drawing a round one, this cell is nothing but a neuron. Here in our chapter, we are talking with respect to the neuron, right? In general, I am writing a cell, but here in our case, this is the neuron. So now observe carefully. For this particular neuron, or in general for this cell, where Na plus is more? Is it present inside the cell or outside the cell? These are standard information under the normal condition. Under the normal condition, you need to know what is the normal. Okay. Please remember, outside the cell, Na plus is more. Outside the cell, Na plus is more. Inside the cell, Na plus is less. Understood or not? That means what a pump will do? Pump will cause movement from low to high. And what a channel will do? It will cause movement from high to low. That's it. These are the points that you should understand. Okay. Next one. With respect to K plus. With respect to K plus is more inside and K plus is less outside the cell. Under the normal conditions, Na plus is more outside and K plus is less outside the cell. Whereas Na plus is less inside the cell and K plus is more inside the cell. This is the general basic information. And now we are talking about Na plus K plus pump. Na plus K plus pump. What it will do? Okay, Na plus K plus pump. Pump is a membrane protein. It is present in the plasma membrane. Okay. And what it will do? This is the pump. Active transport from low concentration to high concentration. Now, for the Na plus, low to high. In this way, it is pumping. For the K plus, it is pumping from low to high. Understood or not? You don't need to by heart these things. If you understand that which one is more on which location, if you understand this, based on this logic, you can write this. Understood or not? So Na plus is moving from low concentration to high concentration. That means it is going out of the cell. K plus is moving from low concentration to high concentration. That means K plus is entering into the cell. But here the thing is, here Na plus molecules are small in size. K plus is large in size. Already in the periodic table you studied, no? So in the group from top to bottom, what happens? The atomic radius will increase. That means overall size is increasing. So that's why what happens is, for the movement of every 3 Na plus out of the cell, 2 K plus are coming into the cell. This is what the basic point is. So that's why what you will say in the Na plus K plus pump, what you will draw. So 3 Na plus are going outside the cell for the entry of 2 K plus ions. This is what you are talking. And that too, this is what about this particular pump. But since it is the pump, it needs energy. So inside the cell, you can understand that ATP is hydrolyzed to ADP plus Pi. This is what exactly happening. And this entire information I have already discussed in the cell chapter in the regular classes, right? So now you need to use it simply. So this is the concept that is related to Na plus K plus pump. So here, with respect to our concept of nerve impulse and generation, you need to understand one point. What is that point? When Na plus K plus pump is working, when it is working, then what happens? Three positive charges, three positive ions are going outside, but only two positive ions are coming inside. Three are going outside, two are coming inside. That means what is the overall net change you are observing? Overall thing is one positive is going outside. One positive is going outside. That means from the inside, positive is lost. Positive is lost. So it will help in getting negative inside. Positive is going outside means inside it is getting decreased. Negativeness is increased inside the cell. So to increase the negativeness inside the cell, this pump is useful. I will tell that how this negativeness is useful in the original mechanism. So this is the overall impact that you need to understand. Since 3 plus are going outside and 2 plus are coming inside, what is the overall conclusion? It is the loss of, overall thing is loss of one positive from inside to outside. This is what you need to understand. This is how Na plus K plus pump is working. So with this pump concept is completed. Now we are going with the channels. In the channels, first we will talk about leaky channels. First, we will talk about the leaky channels. Observe the concepts very carefully. What these leaky channels will do? Suppose this is the plasma membrane. Okay. Now here Na plus is more, and here Na plus is less. Na plus is less. And here inside the cell K plus is more. Inside the cell K plus is more. Outside the cell K plus is less. Okay. And now there are some channels, different types of channels we studied: leaky channels, ligand gated channels, and voltage gated channels. First I am talking about leaky channels. What are these leaky channels? This is the leaky channel for Na plus. This is the Na plus leaky channel. And this is the K plus leaky channel. K plus leaky channel. So you know what is a channel. Channel is a membrane protein that is involving in passive transport. So it helps in movement of the ions from high concentration to low concentration. So you understood this. Based on this, you can understand that. So here from more to low. This is what the function is. Up to this you understood, but you need to understand what is the meaning of this leaky. Why you are using this as a leakage? What is this leakage? You just imagine that suppose it is a pipe. And through this pipe water is passing through. It is flowing in this. Suppose there is a hole here. Then what happens? Majority of the water is going in this direction, but small amount of water is leaking from this continuous. Yes or no? So why this leakage is used here? The meaning of leakage here is these are always open. These are always open. Please understand here. The meaning of leaky here is these channels are always open. That means whether a neuronal cell is in polarized state, depolarized state, repolarized state, or hyperpolarized state, whatever is the condition of the neuron, these channels are always open. Please understand this. These are always open. Whatever is the condition of the cell, these are always working. Since these are always working, always they are trying to move the ions from high concentration to low concentration. That's it. So since these are always open, always working, this type of channels are called as leaky channels. This is what the significance of these leaky channels. Actually, these leaky channels are not playing much role during depolarization, repolarization, hyperpolarization. During these three stages, their role is very less. But they play very significant role during bringing the neuron from hyperpolarized state to resting state. That's why to get the normal resting state, these leaky channels are very important. So this is the point that you need to understand very clearly. Clear? So that is about the leaky channels. Next one. With this first one, leaky channels is completed. Now next point is related to ligand gated channels. Now come to this NCERT diagram. This is the ligand gated channel concept. What is this? Here I will tell you some names, so please observe carefully. This is the axon of one neuron, okay? And the axon of this neuron is branched, and here it is having, at one branch, it is having this bulged position. And this bulged portion is called as a synaptic knob. It is called as synaptic knob. And inside this synaptic knob, it is having some vesicles. And these vesicles are called as synaptic vesicles. Inside the synaptic vesicles, it is having some chemical molecules, and these are called as neurotransmitters. This is this upper part. And for this upper part, there is one more thing. This is the plasma membrane of the axon. Yes or no? And this is the plasma membrane. And the plasma membrane of this axon terminal is called as presynaptic membrane. It is called as presynaptic membrane. Clear? And after this, here there is a small gap. There is a small gap, and this gap is called as synaptic cleft. Synaptic cleft. And after this, actually it is the plasma membrane of next neuron. Plasma membrane of next neuron. This is one neuron, and this is the second neuron part. And what is the second neuron? This is actually the part of dendrite of second neuron. In general, in the normal multipolar neuron, what you are talking? This is the axon terminal of first neuron, and this is the dendrite of next neuron. Understood or not? So this plasma membrane of dendrite of next neuron is called as a postsynaptic membrane. So in this way, plasma membrane of first neuron's axon is called presynaptic membrane. Dendritic plasma membrane of second neuron is called postsynaptic membrane, and the small gap between these two is called as synaptic cleft, and all the three combined it is called as a synapse. Clear? Now observe carefully. Same thing I am telling you. So please observe that concept is also completed. Ligand gated channel concept is also will be completed. Both of them will be completed at the same time. So observe carefully. Now we are talking about ligand gated channel. And here how exactly it is working. Here I am taking some examples for your understanding purpose. Using that example, I will try to explain. Okay. First we are talking about one neuron. This is the cell body. These are the dendrites like this. Okay, this is the axon of first neuron. Okay, I am drawing just rough diagram, so please observe carefully. Okay, so this neuron, this is the neuron that has dendrites. Dendrites received the information. From the dendrites, it has gone to the cell body. From the cell body, the information is coming to the axon. It is slowly coming and it is reaching the end of this particular axon. This is the axon terminal. And this axon terminal is having a bulged appearance. It is called as synaptic knob. Synaptic knob. Clear? Inside this synaptic knob, it is having some vesicles. It is called as synaptic vesicle. And inside the synaptic vesicles, it is having neurotransmitters. It is having neurotransmitters. I am not writing full names. Simply in short, I am writing. These are the neurotransmitters. This is for one neuron. And now this neuron is connected to next neuron. For the next neuron, now we will draw the diagram. This is the next neuron. Okay, and this neuron is also having like this. Okay, and this is the dendrite, right? Actually these dendrites are actually branched like this. This is how we are drawing this, but originally if you are observing, please observe here, actually it is like this. For only one I am drawing like this. Actually it is a branch like this. It is a branch like this, so you can understand that in the same way all these are branched, but for generally for rough diagram we are simply drawing like this. Okay, now you just observe this carefully. This is the membrane of axon of first neuron. It is called presynaptic membrane. Presynaptic membrane. Okay, and this is the plasma membrane of dendrite of second neuron. Yes or no? And this is called as post synaptic membrane, and in between these there is a gap, and this is called as synaptic cleft, and all the three combinedly called as synapse. This is what they are talking. That diagram, actually this portion whatever they have drawn, actually it is this particular part. This is the part that they have drawn. This is the part that they have drawn. Okay, originally this is what the complete diagram is. This is what they are saying with respect to that. And now we are going with the concept. I will draw one more rough diagram. So please observe it very carefully, very carefully. If you understand this, now your concept will become very easy. Observe carefully. First neuron, and this is the axon of first neuron, and it is having this synaptic vesicle. It is the synaptic knob. Inside this synaptic vesicle is present. Inside this neurotransmitters are present. Clear? And for our understanding purpose, we are taking an example of acetylcholine. ACH is acetylcholine. With respect to acetylcholine I am talking, because in the nerve impulse generation and conduction we are talking with respect to acetylcholine. That's why I am talking about acetylcholine directly. Okay, let us consider that this neurotransmitter is acetylcholine. Different types of neurotransmitters are there. Okay, so this is the acetylcholine. Nice. Now what we are saying? Now we are saying that this neuron has received information from where? Through the dendrites. From the dendrites information is coming to cell body. From the cell body it is coming to the axon. And this information is reaching the axon terminal, that means synaptic knob. And now what happens is calcium ions will enter into the cell. Because of this, what happens? Calcium ions will enter into the neuronal this synaptic knob, and now this will help in movement of these particular vesicles towards the presynaptic membrane. So that what happens? Now this synaptic vesicle is slowly moving, and finally it will fuse with the presynaptic membrane, and inside this is the neurotransmitter. After fusing, what happens? Now this is released, these neurotransmitters are released by exocytosis. This concept also I have explained very clearly in the cell chapter. Endocytosis, exocytosis I have explained. So this will fuse. Now the plasma membrane, now it will be rearranged. Now it is releasing this, giving the space, so that now this neurotransmitter called acetylcholine is released into the space. Okay, now observe this diagram very carefully. Okay, now here this neurotransmitter I am making some round molecules. The neurotransmitter I am representing as some round molecules. Observe carefully. So in this way, into this cleft it is released. Now what you are observing? Now we are saying that now this is the plasma membrane of the dendrite. Now it is the plasma membrane of the dendrite that you are talking. Now this is the next neuron. It is like this. Now it is like this. Okay, I hope now you understood this. We are focusing on this particular part. We are not worried about the remaining diagram. Now observe carefully. Now here what you are saying? Here you are saying that on this postsynaptic membrane you are finding some receptors. Observe carefully. If you observe NCERT diagram, these receptors they have mentioned only on postsynaptic membrane. These receptors are not present on presynaptic membrane. Why they are not present? If receptors are present on presynaptic, what happens? Information can go back again. That should not happen. Information transfer should be always unidirectional. It is always unidirectional. That means these receptors are present only on postsynaptic membrane. They should not present on presynaptic membrane. Okay, and here exactly what is this ligand, how these ligand gated channels will work. Now we are talking about this. This is the receptor they are talking. Nice. Actually, attached to this receptor there is a channel. There is a channel that is attached to this. Observe carefully. Okay, and please remember here this neurotransmitter is nothing but acetylcholine. And this, what is this receptor? It is the receptor for acetylcholine. And what is this channel? It is a ligand gated. What is this ligand here? In this case, what is that ligand? It is the acetylcholine. That acetylcholine itself is called as a ligand, and that will help in gating, gate opening and gate closing. Okay, so ligand gated. What type of channel? It is a sodium channel. It is a ligand gated sodium channel. Directly I am going with the original concept. So please remember all these things. When we are studying about the original mechanism, I will directly use these names. So please understand this. So acetylcholine from this synaptic knob is released into the synaptic cleft. Very good. Now it is available in the synaptic cleft. On the postsynaptic membrane of dendrite, we are observing a receptor for the acetylcholine, and attached to this receptor there is a channel, and these channels are called as ligand gated channels. And please remember the concept carefully, please observe it. And this channel is not always open. Please remember, leaky channels are always open, but this ligand gated channel is not always open. That means you need to know when it will be open, when it will be closed. It is not always open means sometimes it is closed, sometimes it is on. And when it will be on, when it will be off. That point you need to understand. Please observe carefully. The point is, this is the receptor. Receptor is not attached to the ligand. Receptor is not attached to the ligand. When receptor is not attached to the ligand, with respect to this acetylcholine sodium channel I am talking, it will be closed. Observe carefully. Observe carefully here. When the receptor is not attached to the ligand, then this channel is closed. It is closed. Even though channel is present, since it is closed, it will not allow movement of sodium ions. It will not allow movement of sodium ions. So in this way, it will be in off condition, switched off condition. Understood or not? So in this way, normally it is in off condition. So when it will be switched on, when the gate will be open, now understand carefully. When the gate will be open, when this acetylcholine, this is the acetylcholine, right? This is the acetylcholine. When the acetylcholine, which is acting as a ligand, this is nothing but a ligand. In our case, this is the ligand. Acetylcholine is the ligand. So when this ligand is attached to its receptor, specific receptors are there. When it is attached to its own receptor, then what happens? Receptor structure will be changed. Once the receptor structure is changed, now the channel structure also will be changed. Since channel structure is changed, now what happens to this channel? Channel will be open. Channel is open. Now the gate is open here. Initially gate is closed, but now the gate is open. So when it will be open? As far as this ligand is attached to the receptor, until then this gate will be open. So once this gate is open, now what happens? Observe carefully. What happens? What type of channel is it? Na plus channel. Channel means without using any energy, from high concentration to low concentration it will come. Where Na plus is more? Outside the cell, Na plus is more. So outside the cell, Na plus is more, and through this it is entering into the cell. From high concentration to the low concentration means it is a channel. It is a membrane protein. Without using any energy it is bringing, but this opening and closing will be decided by the attachment of ligand to the corresponding receptor. So please observe carefully. Once again I am giving the conclusion. With this part will be completed. When the ligand like acetylcholine is attached to its receptor, then the channel attached to this called ligand gated sodium channel will be open, so that it will cause the entry of sodium into the neuron, inside the neuron. So plus is coming into the cell. You need to talk everything with respect to plus and minus. So observe carefully here, plus is entering into the cell. Clear? Nice. This is one point. Now, how long this will be open? How long it will be open? So until this acetylcholine is attached to the receptor. Until then this will be open. So now you need to understand another point. Now, is this acetylcholine permanently attached to this receptor? No. Acetylcholine is not permanently attached, because in this particular cleft there is an enzyme that is called as acetylcholine esterase. Acetylcholine esterase, there is an ester bond breaking enzyme. So that will come and break this particular acetylcholine. So once this acetylcholine is broken, now what happens? Now it will be removed from the receptor. Please observe carefully. Once this acetylcholine is removed from the receptor, then what happens? Now this gate will be once again closed. Once it is closed means there is no further entry of sodium. So in this way, ligand gated channels will help in movement of these particular ions, whatever it is. So in this way they are helping. But this opening and closing of channel will depend on attachment of that particular ligand to the corresponding receptor. This is what the mechanism is. Is it clear? So in this way, this ligand gated channels will work. Ligand gated channels will work. I hope now we understood about ligand gated channels. Come to this concept. Same is the concept here. Nerve impulse is reaching this axon terminal. Now here calcium ions are entering into the synaptic knob, and these synaptic vesicles are slowly moving and they are fusing with presynaptic membrane, and then by exocytosis they are releasing these chemicals called neurotransmitters into the synaptic cleft, and these neurotransmitters, they will bind to the receptors present on postsynaptic membrane. Yes or no? These receptors are not present here. If they are present in presynaptic, again once again it will go back. So that should not happen. That's why receptors are not present here. They are present only on postsynaptic membrane. So now this attachment is causing the opening of some channels, so that now this will induce, now it will trigger the information or the impulse generation in the next neuron, and now it will be going on. So in this way it is keep on going from one neuron to next neuron. So in this way, at these junctions, this is what happening. At the synapse, this is what the mechanism that is happening. So in this way, transmission of impulse from one neuron to another neuron is taking place like this. This is what the diagram is. I hope now you understood this diagram also. This concept also we have completed. Clear? This is what about the ligand gated channels and their action. Clear? Now come to the third one. Voltage gated channels. What is the third one? Voltage gated channels. This concept is very important. First two are little bit easy to understand, but the third concept called as voltage gated channels. Voltage gated channels. Voltage gated channels. How do they work? You need to understand this. These are also channels. These are also channels. In the first type of channels, we said that they are always open. In the second type of channel called ligand gated channel, we said that they are not always open. They are undergoing opening and closing. And who will decide? Attachment of ligand to the receptor will cause opening of the channel. And the detach means separation. Detachment of the ligand from the receptor will cause closing of the channel. That's what we studied for ligand gated. But here, who will decide? The gate opening and closing will be decided by whom? It is the voltage. Voltage will decide whether it will open or not. Since you are talking about the voltage, the basic information required for understanding nerve impulse generation, I will complete here itself under the voltage concept. Okay, because there also we are talking about minus 70, minus 55, plus 35 or plus 45 you are talking, and again minus 70, minus 90, you are talking all these, right? All these things I will tell you here itself, so please observe carefully. Let us take a particular cell. This cell, I am talking cell means this is a neuron. Blindly you just remember, in this particular chapter it is the neuron, and this is the cell, right? And here what we are doing is, we are measuring the membrane potential. We are measuring the membrane potential. How actually we are measuring the membrane potential? Please understand these concepts. How actually you are measuring the membrane potential? See, there are some molecules and ions present inside the cell. There are some molecules that are present outside the cell also. Molecules and ions present outside the cell also. Now here the thing is technical, in simple words if you are talking, how many positives are present inside the cell, how many negatives are present inside the cell. Same way here, outside the cell, how many positives are there, how many negatives are present. Add all these things, add all this, add all this. Understood or not? After adding all these, you are getting some value x. And after adding all these things, you are getting some value y here. Understood or not? So in this way, you are getting some value inside the cell and you are getting some value outside the cell. So for inside the cell, we are considering this as x, and for outside the cell, we are considering this as y. Okay. So in this way, some values we got. Now compare these two values. Are these values exactly same? Is x is equal to y? No. If x is equal to y means cell will die. Please observe carefully. If x value is equal to y, cell will die. Please understand this. Anyhow, this concept was there in previous NCRT in the biomolecules, but now they have deleted, so we are not going into those details. So leave it. So x is never equal to y. Understood or not? If a cell has to survive, if an organism has to survive, these values should not be same. So that means at one location it is high, at one location it is low. Now scientists have calculated these values and they observed that value of x, that means the potential inside the cell, is less than the potential outside the cell. Please observe carefully. The value inside the cell across the plasma membrane, we are considering this. That means inside the cell, outside the cell. Across plasma membrane means what? Inside the cell and outside the cell. So here what is the value they got? Inside the cell always it is lesser when compared with outside. Under the normal resting conditions, if there is no nerve impulse transmission, please understand what is resting means. Nerve impulse is not present at that time, that is the resting stage. So at the resting stage, they measured these values and they concluded that the value of x, that means the potential inside the cell, is less when compared with the outside. So now what you need to write? Value of x is less and the value of y is more. Okay. This is what the ultimate conclusion is. Please observe carefully. Ultimate conclusion is value of x is less and value of y is more. So instead of saying less and more, scientists have adopted a convention. What is the convention they adopted? Since it is less, they used the negative value. Since it is more, they used a positive value. Please observe carefully. This is what originally that negative means. This is negative inside the cell, inside the plasma membrane, it is negative. Negative meaning is what? It is less when compared with outside. That's why under the normal resting condition, what you will write? Sorry, inside negative, outside positive. This is what you are writing. Why you are writing inside negative, outside positive? Because inside the value, total value is less. Outside the total value is more. That's it. Instead of saying less and more, they are using minus and plus. That's it. This is the original concept. Please understand this. Okay. In this way, under the normal condition, they are measuring like this. And how much it is less? So based on this, they are writing this. So that's why you don't need to talk both of them at the same time. When compared with outside, inside how much it is less? It is 70 millivolts, it is less. Please observe carefully. When compared with outside, please observe this point very carefully. Once again I am repeating. When compared with outside, inside it is less. But how much it is less? 70 millivolts it is less. When compared with outside, inside it is 70 millivolts less. Since it is less, you are using minus symbol. When compared with the outside, inside it is 70 millivolts lesser. That's why you are writing this one as minus 70 millivolts. That's why what is the standard? Whenever they are talking about the membrane potential, please observe carefully, membrane potential, always you need to talk inside the cell. Means inside the plasma membrane, inside the cell only you need to talk. Don't talk outside the cell. Is this clear? Because it is the standard convention. When compared with outside, what is the value inside? If it is lesser inside, you just simply add minus. And how much less? Write that one. Suppose it is more inside. Suppose in some cases during the depolarization it can change. Suppose it is more inside. Then slowly increase the value. and then you can start writing the plus. In this way they are adding minus, that's it. Okay, so this is what the original concept. So please remember, the conclusion is, whenever we are talking about the membrane potential, always we are talking inside the cell. So what is the resting membrane potential? At the rest means when there is no nerve impulse, what is the membrane potential? It is minus 70 millivolts. So that's why minus 70 millivolts is considered as resting membrane potential. Is this clear? Now we are going with the concept slowly. Please remember, I don't want to write the original information here, but almost original information I will try to bring up here. Okay, observe carefully here. Now here inside the cell it is minus 70 millivolt. Clear? Now we will say that through some process, I will tell you later, through some process, suppose some positive ions are coming inside. Positive ions are coming here. Add one positive to this. This is minus 70. To this, one positive is added. That means what happens? It will become minus 69. For understanding purpose, I am saying. So minus 70 plus 1 means what? Minus 69. And further plus is coming. Minus 68, minus 67, minus 66, minus 65. Do you able to understand this? That means when plus is coming inside, what happens? Now this value is keep on decreasing. Minus 60. Minus 50, understood or not? Minus 40, minus 30, minus 20, minus 10, and then 0, 0. And if pluses keep on coming, then what happens? It will go to plus 10, plus 20, plus 30, plus 40, plus 45. Do you have to understand this? So in this way, please understand, at the resting condition it is minus 70 millivolt. Suppose some positive ions are coming into the cell. If positive ions are coming into the cell slowly, these values inside the cell will be slowly changed. It all depends upon how much positive ions they are coming. If more and more positive ions are coming, it can go like this also. These are the things that you need to understand. Yes. Suppose. So in this way, positive ions are coming into the cell. That's why it is changing like this. If positive ions are going out of the cell, suppose it is plus 45. Now positive is going out of the cell. If positive is going out of the cell, now what happens? Slowly, plus 45 coming, plus 40, plus 30, plus 20, plus 10, 0, minus 10, minus 20, minus 30, minus 40, minus 50, minus 60, minus 70. Understood or not? And if even after getting minus 70 also, still plus is going outside. Still plus is going outside. Now what happens? It will become minus 8, it will become minus 90. Like this, the potential inside the cell we are measuring. So please observe carefully. Potential inside the cell we are measuring. At the resting stage it is minus 70 millivolts. Entry of positive ions into the cell will cause the decrease in this value from minus 70 to minus 60, minus 50, likewise 0, and then plus 20, plus 30, plus 45, likewise. If positive ions are going out from plus 45, it is going to plus 40, plus 30, plus 20, plus 10, 0, minus 10, minus 20, minus 30, minus 40, minus 50, minus 60, minus 70, and even still a plus are going, then it can go up to minus 80, minus 90 also. So these are the changes that you are observing in this particular mechanism of nerve impulse generation and conduction. So these are the some potential changes. I hope now you understood how exactly these potential changes are taking place. You should not get this doubt while I am explaining the concept. Okay, so please understand this. In this way, potential changes are taking place. Now it is the concept of voltage gated channels. Voltage gated channels. Please observe carefully. Voltage gated channel. In short, I will write this as VGC. So voltage gated channel for Na plus. Voltage gated channel for Na plus. Please observe carefully. When it will be open? Please remember here, when the value is minus 55 millivolt or less, less means what? In this case, less means it is not minus 56, minus 57. Less means minus 54, minus 53, minus 52, minus 51, likewise. And this is the threshold value. Please understand this. For the opening of voltage gated channel, you need minus 55 or less than this, like minus 50, minus 40, anything is okay, but not more than minus 55. Is this clear? So this is what is the requirement, and it is open till plus 45 millivolts. Please observe carefully. So here in this way, what you are saying here, now understand this one, minus 70, it is keep on coming up to minus 55. Once it is coming to minus 55, voltage gated channel for Na plus will be open. So that what happens? Now voltage gated channel, now who will decide this one? This gate will be decided by, now this is the gate, and till now it is closed. But once it is minus 55, now what happens? This gate will be open. This gate is open. Now what happens? Now Na plus channel means what? Higher concentration to lower concentration. Na plus is more outside, right? Now higher to lower, now it is going. Now since plus is added, now what happens? Minus 55, minus 50, minus 40, minus 30, minus 20, minus 10, 0, plus 10, plus 20, plus 30, plus 40, plus 45. Generally up to plus 45 it is going. Once it is going to plus 45, voltage gated channel will be closed for NaCl. Sorry, for Na plus. Sodium voltage gated channel will be closed at plus 45. So in this way, who will decide opening and closing? Observe the concept. Who will decide opening and closing of this channel? It is the voltage. It is the potential. It is the potential difference across the plasma membrane that will decide whether this particular channel should open or not. Do you able to understand this? This is what about the voltage gated Na plus channel. Now what happens? At plus 45, voltage gated Na plus will be closed. Once it is closed, now what happens? At this particular one, voltage gated channel for K plus will be open. Please observe carefully. It is bringing from minus 55 to plus 45. Who is bringing? Voltage gated Na plus. Then this will be closed. Once it is closed, to bring back to normal minus 70, what should happen? Voltage gated K plus is opening. So when it will open? At plus 45. So that what happens? K plus will come out of the cell. Plus is going out of the cell. Once plus is going out of the cell, what happens? Now this value will decrease. Yes or no? Decrease means technically it will increase. So plus 40, plus 30, plus 20, plus 10, 0, minus 10, minus 20, minus 30, minus 40, minus 50, minus 60, minus 70. It should stop at minus 70, but it is still not stopping. So it is going up to minus 90 millivolt. At minus 90 millivolt, voltage gated channel for K plus will close. Please observe carefully. Actually resting is minus 70, but still these are not closed. It is going up to minus 90. Understood or not? Now what should happen? Now here what should happen? To bring with, now at minus 90 what happens? Voltage gated channel for K plus will be closed. Okay, nice. And now from minus to 90, it should come back to minus 10, minus 70, because this is the resting state. So here what happens is, now again there is entry of this K plus into the cell and movement of Na plus out of the cell. Please observe carefully. And what are these? These are leaky channels. Sorry, it is the reverse, right? So it is more inside, right? Reverse. I am sorry. So here what happens is, so K plus from inside it is going outside, Na plus outside it is coming inside. These are the leaky channels. And here is the pump. Here is the pump. 2 K plus going inside, 3 Na plus coming outside. It is the pump. Please remember, these two are very important. From minus 90 to minus 70 millivolt, it should come back. And for this particular process, leaky channels and pumps will play very, very significant role. Leaky channels are present even in this condition also. But their role at that time is very less, because voltage gated Na plus is opened means it is like a opening of a dam gate. And this leaky channel is like a normal small canal. The amount of water going in that small canal will be very, very less. But this is like opening of a dam gate. Now you can imagine how much of water will go. In the same way, in front of this, the contribution of these leaky channels is very, very negligible. That's why here we are not talking much about leaky channels. But here at this condition, Voltage gated Na plus is closed, voltage gated K plus is closed. So who will play a significant role now? Now leaky channels will play very significant role along with pump. Anyhow, pump is ultimate. That is very important. Okay. So this is how exactly the membrane potential will be changed. So these are some things that we are actually talking during this nerve impulse generation and conduction. I hope now we understood this. Anyhow, step by step I will explain. Don't worry. This is just basic idea with respect to voltage gated. So in this way, there are some channels, but the opening and closing of these channels will be decided by the voltage. That's why they are called as voltage gated channels. So in this way, we have studied about this leaky channels, ligand gated channels, voltage gated channels. All these are very important to understand this concept. Along with this, we are talking about Na plus K plus pump. These are some important things that we need to understand very clearly before we are going with the concept of generation and conduction of nerve impulse. Okay, we understood these things, and this table, this graph also I will explain, don't worry, and this point also we have completed. So once we complete this, we can go with the brain and all, but this point is very important. I will take 5 to 10 minutes for the explanation of that part, because I need to explain slowly, right? Because many of you don't know about these things. So now we will go slowly. Observe these key things. I will draw several diagrams at the same time, so observe very carefully. First thing is, suppose there is a neuron and these are the dendrites, okay, and these dendrites received information and they are passing to the cell body. From the cell body it is coming to the axon, and this axon is having axon terminals, and they are having the synaptic knobs, and inside these synaptic vesicles, inside these neurotransmitters are present. Clear? And now we are talking about the next neuron. This is the next neuron. For this next neuron, these are the dendritic membrane, suppose. Okay, on this dendritic membrane, there are some receptors for this neurotransmitter. Once this impulse is coming up to this, now calcium ions are coming into the cell. Yes or no? Not in all the cases, we are talking for acetylcholine, so that's why I am saying about the calcium entry. So a calcium is entering into this. This vesicle will come and fuse with this plasma membrane, and the neurotransmitter is released into the cleft. And once neurotransmitter is released into the cleft, now neurotransmitter will bind to the receptor. Clear? Keep that point there, and then now I will draw one graph. Observe carefully here. In this graph, I am mentioning about minus 90. All these are in millivolts. Okay, this is the minus 80. This is minus 70 millivolt, and somewhere here we will write minus 55 millivolt. Somewhere here we will write 0, and somewhere here we will write plus 45 millivolt. Okay, these are some important values that we need. Okay, anyhow, 0 is not needed, but still I am writing like this. Clear? Now, how exactly we are drawing the graph? For the normal neuron, this is the second neuron. We are talking about the second neuron. Inside this second neuron, how exactly nerve impulse will be generated? Till now, impulse is present in this neuron. From this neuron, now it should come into this. Now, in this neuron, how nerve impulse will be generated first? First, you need to talk about how exactly it is generated. Once generated, how it will be conducted in the neuron. These are the things that we need to study. And once you know up to this, again at the end, the same process will occur for this one also. Yes or no? Now here it is connected to the next neuron like this. So this process is same. Whatever is happening here, same is happening here. But within this neuron, what exactly happening? That we need to observe carefully. Now, till now, before attachment of this neuron is in resting stage. What is this resting stage is called as? Resting stage is called as polarized state. Polarized state is the resting state. That means at this particular state, nerve is, or means neuron is not transferring any information. That's why it is called a resting stage. So at the resting stage, I am drawing the diagram. So please observe carefully. Okay. At the resting stage, we are saying that inside it will have negative, outside it will have positive. This is what the resting membrane, right? And what is the value of resting membrane potential? It is a minus 70 millivolt. Always we are talking inside the cell. So inside negative, so it is the minus 70 millivolt. Clear? And here we are writing about some things. Observe this carefully. First one is regarding the Na plus K plus pump. Na plus K plus pump. Please observe carefully, one by one I am writing, observe carefully. Next one is the voltage gated channel, VGC, voltage gated channel for Na plus. Next one is voltage gated channel for K plus. Voltage gated channel for K plus. Understood or not? Next one is the leaky channel. Leaky channel for Na plus, leaky channel for K plus. This is for Na plus, this is for K plus. These two are leaky channels. Leaky channels. Please observe carefully. For the leaky channels, I did not close this. These are open because leaky channels are always open. So that's why they are open. And this is the resting or the polarized state. Why the resting state is called polarized state? In the resting state, there is a polarity. There is a polarity across the membrane. What is this polarity? Outside positive, inside negative. Outside positive, inside negative is the polarity that we are observing in the resting state. That's why it is called as polarized state. And the next important point you need to understand is, during this resting stage, who will work? Who are actually working in the resting stage? Please observe carefully. Pump is working here so that it is sending 3 Na plus outside and it is bringing 2 K plus inside the cell. So this tick mark means it is working, pump is working. And what is this one? Voltage gated channel. See the diagram, I have kept it as a closed. So into mark means, or cross mark means not working. It is not working. Voltage gated channel is not working for Na plus because what is the voltage here? Minus 70. When this will be open? This will be open at minus 55 millivolt. And voltage gated K plus, where it will be open? It is open at plus 45 millivolt. But here it is minus 70. So these two are closed. Leaky channels are always open. So what is their function? Na plus will come from higher to lower. K plus is moving from higher to lower. Na plus is entering into the cell. K plus is going outside the cell. It is very less. Very less. And again between these two, if you observe NCRT wording, what they give? Under the resting condition, if you are observing, these leaky channels for K plus, they are actually number wise. If you see, suppose this is the axon membrane, on the membrane you just see count the number of channels. Actually for K plus, more channels are present, more number of channels are there. For Na plus, they are very less. That's why what they say is resting membrane is more permeable to K plus, and resting membrane is almost impermeable. Please remember, they said almost impermeable. They did not say completely impermeable. They said almost impermeable means very less permeable to Na plus, because the number of Na plus channels are very less. That's why it is very less permeable or almost impermeable to Na plus. This is the meaning, because they are talking with respect to leaky channels. But when compared with this, it is more permeable to K plus because the number of K plus leaky channels are more. That's why it is more permeable. So please understand this. What is the ultimate conclusion of this? The plus, Na plus, the plus coming into the cell here is less, but the plus that is going out of the cell is more. Remember this particular logic. I will link it up at the end of this particular diagram for this particular slide. Okay, remember this. And what is the contribution of the pump? Pump is also doing the same thing. It is sending 3 plus outside, 2 plus into the cell. What is the overall contribution? Overall, if you see, plus is lost, plus is going outside. Yes or no? 3 going outside, 2 coming inside means what? One overall value is 1 plus is going outside. That means here also plus is going outside. Here also plus is going outside. This is the conclusion. You need to remember the conclusions. Why it is happening like this? Because from the hyperpolarized, you need to bring it back to normal. That's why it is doing like this. I will link up all the points. Please observe carefully. This is a polarized normal condition. Clear? At the normal condition, what is the value it is? It is minus 70 millivolts. This is the minus 70 millivolts, and this is called as a resting. This is the resting potential. This is the normal resting potential. And when there is an impulse, and how this impulse will come, now observe the concept carefully. This is what the concept is. Already I have explained you, so please, directly I am going with the conclusion, so please observe carefully. Now here what we said, Suppose this is the plasma membrane of the dendrite. Okay. Now what I said, from this acetylcholine is released into the cleft, and here is the receptor, and now this acetylcholine is attached to receptor. When acetylcholine is attached to receptor, sodium gated channel will be open. I told you this or not? So when acetylcholine is attached to the receptor of postsynaptic membrane, then Na plus ligand gated channel attached to this will be open, so that what happens, Na plus from the outside is entering into the cell. Na plus from outside is entering into the cell, so plus is entering into the cell. Initially it is minus 70 millivolt. Now Na plus is entering. Now what happens? Now this value will come to minus 69, minus 68, minus 67, minus 66, minus 55. Do you have to understand this or not? So in this way this value is keep on coming. So when this value is keep on coming like this, minus 64, minus 63, minus 62, minus 61, minus 60, minus 59, minus 58, minus 56, minus 57, minus 56, minus 55. Please understand this. So from this it is coming up to this point. Understood or not? And this is called as a threshold. This is called as a threshold. What is the meaning of this threshold? This is the minimum potential that is required to generate the action potential. To generate the action potential, the minimum potential that is required is called as the threshold value. What is the meaning of this? Suppose Na plus is keep on coming here. From the minus 70, it came up to minus 60 only, And then this acetylcholine was removed, and then this gate was closed. That means it came up to minus 60 only. So if it is coming up to minus 60, can it generate the action potential? No, it cannot generate. So please understand what is the meaning of this threshold and what is its significance. To generate the action potential, this initial entry of Na plus into the cell, it should bring this value to minus 55. Then only once it is coming to minus 55 or lesser than this, means minimum of minus 55. It can be minus 54, minus 50, minus 40, anything is okay. But minimum minus 55 is required. Then only action potential will be generated, or else there is no action potential generation. That's why all these things are called as all or none principle. Generated means complete action potential will be generated. None means it is not generated at all. In this way, it is like all or none principle. So that's why those which are crossing threshold, they only will form the action potential. Those which are not crossing threshold, they cannot form the action potential. That's why this should be open till it is becoming at least minus 55. If it is minus 50, minus 40, that is well and good. So at least minus 55 should be happening here. So until then it should be open. Suppose it is there until minus 55. So now it came up to minus 55. Now what happens? Already I told you. So once it is coming up to minus 55, then what happens? Voltage gated channel for Na plus, where it will open? At minus 55 it will open. So now it will start opening. Once this voltage gated channel for Na plus is start opening, now what happens? If VGC all from minus 70, it came up to minus 55. Now voltage gated channel for Na plus is opening. Now what happens? Now more and more plus is coming into this. Now more and more plus is coming means from minus 55 it will go to minus 50, minus 40, minus 30, minus 20, minus 10, 0, plus 10, plus 20, plus 30, plus 40, plus 45, up to plus 45 it is going. And going up to plus 45, that means now this stage is called as depolarized. Why it is called depolarized? Now observe carefully. Now observe carefully. It is going up to plus 45 inside the cell. Inside the cell plus means outside the cell will be negative. That means when compared with outside, in this particular state, excited state, it is called excited state, inside it is becoming more when compared with outside. That's why inside it is becoming plus. Now this is called D. D means what? Removal. Removal of polarity. Depolarized state. This is the depolarized state. It is also called as excited state. This is the excited state. Understood or not? This is what. And why it is called depolarized? Now you may think that here also there is a plus and minus, that is also called a polarity. No, we are not talking like this. What is original polarity? In the resting state, outside positive, inside negative is called as the original polarity. This is the original polarity. Is the original polarity present here? No. Here original polarity is lost. D is the removal of polarity. Which polarity is lost? Original polarity, that is outside positive, inside negative, is lost. That's why you are getting like this. So that's why outside negative, inside positive is called depolarized state. It is the depolarized state, loss of polarity. And here it can go up to minus, as per standard information we are writing plus 45, but in different case it can go even up to plus 60, plus 70 also it can go, but plus 45 is enough for our studies. This is what happening. But now you need to understand that to get this depolarized state, now who is working here? Just now we stated that voltage gated channel for Na plus is open. I'm not writing, these are the things I'm writing, because every time if you are writing, so space will be a problem. So voltage gated channel for Na plus, VGC Na plus is open. I will write inside the cell, no problem. Okay. Next one, please remember this is the cell, right? This is the cell. Don't get confused. Don't get confused between these lines. You need to understand this very clearly. Okay, I will make a round like this. It will be easy for you. Next one. See, when it crossed this minus 55, voltage gated channel for Na plus has opened. So that's why because of this what happened? Na plus entered into the cell. That's why we got this plus 45. Yes or no? This is what they are saying. And now at this particular stage, means from the polarized to depolarized state, when it is at the resting, this is working. But once it is crossing that minus 55 threshold, once it is crossing minus 55, what happens to this pump? Now understand the meaning. What actually is needed? Inside positive is needed. But what is the mechanism of the pump? Pump is actually sending the plus outside. But here you need more positive inside the cell. That means during this depolarization state, pump should not be active. So pump is inactive. Pump is not working during this depolarization process. During the depolarization process, pump is not working because pump is actually trying to remove positive from inside. But actually we need positive inside. So that's why pump is not working. So you can understand these mechanisms. That's why you don't need to by heart understand this. So pump is not working. Voltage gated channel, yes, it is bringing a positive inside. Yes, we need. So this is working. But K plus, if K plus opens, it will go outside. So this is not working. So that's why this is still in the closed state only. During this voltage gated channel for K plus is in closed state only. It is not open. It is not working. But anyhow, these leaky channels for Na plus, leaky channel for K plus, they are always open. But their contribution is very, very less in this process, right? So we are not worrying much. Pump will contribute significantly. Voltage gated channels will contribute significantly, but leaky channels during this process, their contribution is very less. So they are, even though they are open, they will not make much changes. So that's why pump should be inactive. Voltage gated channel for Na plus is active. Voltage gated channel for K plus is inactive, and anyhow leaky channels are open. This is what happening during this depolarization state. So in the graph, what we are doing? In this graph, from this minus 55, we are taking it to the plus 45. And this one is called as depolarization. This is the depolarization. Understood or not? And this maximum is called as action potential. This maximum is called as action potential. Understood or not? And this is what happening because of the initial entry of this Na plus, this voltage gated channels are opening, so that now more and more Na plus is coming. Now in a particular segment, please remember, in a particular segment, now you need to understand another concept. What is this? We are talking about the neuron. Suppose this is a long neuron, right? Suppose for the complete neuron as a straight line I am drawing, this is a long neuron. Generally, many students, they are thinking that in the entire neuron at a time this potential will be generated. No, this is very long neuron, that much potential cannot be generated in the entire neuron. So actually it is happening in a segment by segment it is keep on happening. Understood or not? Suppose this is the segment A, segment B, segment C, D, E, like this. Okay, and whatever the process we are studying here, though I am making this much long, actually it is in this segment only. In this segment only we are talking about Na plus K plus pump, Na plus K plus pump, voltage gated channel for Na plus, voltage gated channel for K plus, and then leaky channels. All these things we are studying in this segment only. In this way, in the entire neuron, all these are present in this way. Now observe carefully. Initially here what was there? Initially here inside negative, outside positive, inside negative, outside positive, inside negative, outside positive. This is the resting. This is the resting, right? This is the resting. Inside negative, outside positive. But because of this process in one segment what happened? Inside positive and it became outside negative. Yes or no? That means only this segment got depolarized. Only this segment got depolarized. But remaining all these are in resting state. All these are in resting state. Observe the concept carefully because inside negative, outside positive. This is normal resting. So in this way, first one segment is undergoing depolarization so that inside it will create plus 45 millivolts. Understood or not? So in this way, initially in the fresh neuron, because of the entry of this, initially it is creating the threshold, and once this threshold is achieved, now depolarization is occurring. This is the initial step. Once one segment got depolarized, now this value of plus 45 is enough. Now again there is no need of one more ligand gated, not needed, and this will induce depolarization in the next segment. This potential will induce depolarization in the second segment. So in this segment, now once this got depolarized, now it will induce in the next segment. And in this segment what happens? Now here also once again voltage gated channel for Na plus will open and K plus Na plus pump will be closed. Voltage gated channel for K plus will be closed and leaky channels are open. So once again in this segment also, now it will become depolarized. Now here what happens? Inside positive, outside negative will occur in this segment also. That means this is also depolarized. This is also depolarized. Observe the concept carefully. So for the initial depolarization, that ligand gated channels are important. Once one segment is depolarized, now this will induction. This will induce the depolarization in the next segment. Now observe carefully. Now it got depolarized means, now what is the current movement? Whenever we are talking about the current movement, it is about the movement of the positive charges. So inside this positive is going like this. But initially what happened? Here the positiveness is coming like this. Yes or no? Because once this segment induced the depolarization in the next segment, now this will come back to normal. This will come back to normal. Please understand this. So that means once again it will become outside positive, inside negative. So please understand this. What is the purpose of this depolarization? To induce depolarization in the next one. That means action potential from this is going to this. Once it got depolarized, it will induce in the next segment, so that it will wait until it got depolarized. Once it got depolarized, now again it will come back to normal, and now this will induce in next one, this will induce in next one. So in this way, segment by segment, the same steps of depolarization will occur. Understood or not? So in this way, for the depolarizations we are studying here. So the depolarization in this one will induce depolarization in this. Depolarization in this will induce in the next one, in the next one, in the next one. In this way, the action potential from this is going to this. In this way, segment wise it is keep on moving one after the other. This is how exactly the nerve impulse will be conducted. Understood or not? Nerve impulse is keep on conducting. Nice. It is keep on going until the end. Once it is going to the end, what happens in the next neuron? You know, in that neurotransmitter will be released, new ligand gated channel will be open, and there also it will be keep on going. Fine, nice. So in this way, up to depolarization it is completed. But once this segment induced depolarization in the next one, this function is completed. Now from this depolarized state, it should again come back to repolarized state. Re means again polarized. Again polarized means what is this? Original condition of inside negative, outside positive should come. Now here you just see this is depolarized, positive inside, negative outside. Now this will induce in the next segment. Once it induced in the next segment, again it will come back to normal, means inside negative, outside positive will come. So coming back to original, that means once inducing in the next one, it is keep on going forward. And this segment should come back to normal. How it will come back to normal? We will see this. How it will come back to normal here? Coming back to normal is called as repolarized or repolarization. Re means again getting polarized. Again getting polarized means, observe carefully here. Again getting polarized means ultimately what should happen? Inside negative, outside positive should occur. So now understand carefully, but here positive is present inside. And how you will get negative here? You are not bringing negative here. What you are doing? You are sending positive outside. How you will send positive outside? Observe carefully. Na plus should not come inside. So that's why Na plus voltage gated channel VGC for Na plus is closed, but VGC for K plus will be open. Okay, once it is getting plus 45, now it will be opened. So once it got open, now what happens? K plus from the cell is going outside. Plus is going outside. From the plus 45, plus is going outside means ultimately you will get minus 70 millivolt. And not only voltage gated K plus, pump is also working because pump is sending 3 Na plus outside and bringing 2 K plus inside. That means pump is also helping in sending one positive outside. So positive sent outside, positive sent outside. And anyhow these leaky channels are working, both of them. Understood or not? So both of them are helping in sending the positive outside. So pump is working, but voltage gated Na plus is not working, and K plus voltage gated K plus is working, and anyhow leaky channels are working. And in this way, these are the things that are happening, so that from the plus 45 we are getting back to minus 70 millivolts. Please understand this. But the thing is, at minus 70, K plus voltage gated channels are not closed. They are still working. So that's why instead of stopping at minus 70, it is going up to minus 90 millivolt. It is going up to minus 90 millivolt. So going from minus 70 to minus 90 is called as hyperpolarized. Originally polarized is what? Inside negative, outside positive. But here we need the charge of say minus 70 millivolt. But it is going more than this. Since it is going more hyperpolarized, it is polarized more than required. Hyperpolarized. It is the hyperpolarized. Okay. So in this graph, what you are doing is coming from this plus 45 to again, this is the normal resting, right? Coming back to normal 70, minus 70 is called as repolarization. But it is not stopping at the minus 70, it is going up to minus 90. It is going up to minus 90. So that's why this is called as hyperpolarization. Okay. And after this hyperpolarization, what happens? Observe carefully. After this hyperpolarization, okay. So now I am drawing one more thing. So now observe carefully. I don't need to draw this one. Next one is this one only, right? After hyperpolarization, what happens? Once again, it will come back to from minus 90 to minus 70. But during this time, what happens? This voltage gated K plus will be closed. Anyhow, this is closed. So both of these are closed. But the thing is, you just understand the problem here. The problem is in the normal cell, once it is coming back to original polarized, what you need to get? Na plus more outside and K plus more inside is required. This is what is required actually. But during this process, what happened? Na plus came inside. It is present inside the cell. And K plus is going outside. Understood or not? But actually, if Na plus coming inside and storing, K plus is going outside and present there, then in the next round, how it will happen? So that's why from the hyperpolarized, it should come back to normal resting state. So that's why what should happen? Here what should happen? Na plus should go outside the cell. The stored Na plus inside this should go outside the cell. The K plus from outside should come inside the cell. Understood or not? That's why pump is important. What pump is doing? The stored Na plus will be sent outside and the K plus present outside is coming into the cell here. So that's why it is again bringing back to normal. Anyhow, these two are not present. And what this Na plus will do? And Na plus channels are very important. Please observe here. And since Na plus is present more here, now this extra Na plus is going outside. Their function is to move from higher to lower. That's it. In this case, inside more Na plus is there. So now Na plus will go outside. And here K plus is present more outside. So that will bring back to normal. So in this way, always they are bringing from higher to lower. It is not from only from out to in or in to out. Wherever they are higher to lower only they are bringing. So in this way, what happens? Na plus is going outside, K plus is coming inside. This is also Na plus is going outside, K plus is coming inside. So overall, what happens once again? Once again, it will become Na plus more outside, K plus more inside. So that now once again this value is coming back to minus 70 millivolt. So once again it is ready. Which is ready? This segment is ready. Initially this segment got depolarized and this depolarization induced the depolarization in the next one. So once it induced in the next one, now again it is coming back to repolarization, hyperpolarization, and then once again normal resting. This segment is coming. And now what about this one? This got depolarized, this will induce in the next one. And again this will come back to the resting. So in this way, segment by segment, it is keep on passing the action potential. Once it is keep on passing the action potential to forward, this segment is again coming back to normal resting. In this way, this segment is coming back to resting again. Once it comes back to resting again, then only it can receive another impulse or another action potential. So in this way, before coming to the resting, if another one is coming, it cannot receive it. So that's why there is a time period that is required, and that time period is called as refractory period. Now let us complete this. From the hyperpolarization, again it is coming back to the normal resting. So observe this one from here to here. Here it has started and again coming back to the normal resting. Clear? So that's why this time period is called as refractory period. It is called as refractory period. And during this period, already it is in a particular process. So when it is in the refractory period, it cannot receive one more signal. Once it comes back to normal resting, then only it can receive another signal. This is what another important point that we need to understand. Clear? So in this way, segment by segment, information is keep on moving forward. Then each and every segment is again coming back to normal resting condition. That's it. So in this way, nerve impulse is generated and it is conducted. This is what the overall information regarding this particular process. I hope now you understood this. Here, this is the segment A. Here what they are saying, initially this segment got depolarized. That's why inside positive, outside negative. But remaining axon or remaining neuron is in resting. Inside negative, outside positive. But what happened? Once it got depolarized, now this is inducing depolarization in next segment. It is inducing depolarization in the next segment, so that the movement of positive charge indicates the movement of current. So inside the cell, positive charge is going here, because in the next segment here it will become plus, outside it will become negative. So in this way, the movement of current inside the cell is from segment A to the segment B. That means from the first segment to the next segment. But the movement of current outside the membrane is from the B to A. That means from second segment to the first segment, because here what happens, one again it will come back to the normal resting, so it will become plus here. So in this way it is coming back. So please remember, the movement of current inside the cell is from first segment to the second segment. Movement of current outside the cell is from second segment to the first segment. These points also you need to observe. This is how this depolarization, repolarization concepts are very, 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 very important, and you need to understand this. This is the normal condition, and here this is the stimulus. Once it is reaching the threshold, then only it will undergo depolarization. This highest is called as action potential, and again that particular segment is inducing the depolarization in the next one, and this segment is again coming back to normal repolarization, but it is going up to this hyperpolarized state, again coming back to the normal resting, and now this particular entire time period is called as a refractory period. During this particular refractory period, now it cannot receive another signal. Once again it is coming back to resting, then only it can receive the another signal. This is what they are saying. Okay, and nerve impulse conduction is always in one direction, dendrites to cell body to axon. In this way only it is going, and since it is a direction specified in one direction and it is going at a particular speed, so please remember, speed, if it is having a direction, then you can call it as a velocity. So that's why the term velocity can be used in this particular case. So what is the velocity of this particular nerve impulse transmission? But anyhow, that is a higher level concept, so leave it. But in this way we are using these particular names. Okay, so resting or depolarized state, repolarized state, hyperpolarized state, and again it is coming back to normal resting state. This is what they are talking, and anyhow this I told very clearly. And in this way, at the junctions, nerve impulse is moving from one axon to the other axon, and in this way this is a chemical mediated. This is the chemical called neurotransmitter mediated nerve impulse transmission. Yes or no? And here we are talking about some different types of synapses called electrical synapses and chemical synapses. In the case of electrical synapses, the neurons are very closely present in such a way that the last segment of first neuron can directly induce depolarization in the first segment of next neuron, because they are very closely present. This is like electrical induction, right? So it can directly induce the depolarization in the next one. So that's why here in the electrical synapses there is no chemical release, there is no neurotransmitter release, directly it will induce, and such type of synapses will help in quick information transfer. But this type of things are very, very rare, present in human, but very, very rare in the human. But majority of the types of synapses are chemical synapses, and in these cases different types of neurotransmitters. Some are excitatory, means they can induce the action potential in the next neuron, whereas some are inhibitory, they will stop the generation of nerve impulse generation. In this way different types are there, but in our syllabus as part of NCERT content we are talking only about the excitatory type, how nerve impulse will be generated in the next neuron. With respect to acetylcholine we are talking. Okay, but there are some others like GABA, gamma aminobutyric acid, that is inhibitory neurotransmitter. If that neurotransmitter is released, that will stop the generation of action potential in the next neuron. In this way different types of neurotransmitters are involved in different mechanisms, they are having different purposes. Clear? This is what about the transmission of the nerve impulse, and anyhow synapse and everything I have explained. So these things are very simple. So with this, the neuron structure, nerve impulse generation, conduction, and then this transmission of impulse, everything is completed. Now we are going with this small part. I am saying very, very small part, because in the NCERT deliberately they gave very less content, but some teachers are unnecessarily they are teaching very, very huge content. Okay, this is very interesting part, I do understand, but if you are keep on teaching in this particular thing, so there is no limit for this, there is a very, very huge content. So my suggestion is, please go through only NCERT sentences, don't go even a single sentence out of NCERT. But here what I did is, there are some sentences given extra in the summary. Okay, and those sentences also I have copied at the corresponding content, so I will also I will read those things as part of that particular content. Clear? Quickly let us go through this. In the central neural system we are talking about the brain and spinal cord, but spinal cord is not there in our syllabus, please remember this. So we are talking only about the brain, and here the brain is the central information processing organ. This is the main one that is receiving information, and it is a processing and then giving back the signals to corresponding organs. So it is central information processing organ of our body, and it acts as command and control system. It is commanding and controlling all the organs and the activities of an organism. It controls the voluntary movements, yes or no? So that that skeletal part is under the control of this one, right? So that's why it is under the, it is also controlling the voluntary movements and also involuntary things, many involuntary things. All these things are under the control of this brain, and some are under the control of spinal cord also. And balance of the body is also controlled by the brain, and functioning of vital involuntary organs like the lungs, heart, kidneys, and all these things. See, under the regulation of all these things, we studied that in the brain there are some organs, there are some portions, and they are controlling the activities of all these things. Many things we study, no? This is what they are saying. And also the rhythms of our body. Okay, so involuntary organs like all these things, thermoregulation, hunger, thirst, and circadian rhythms of our body, activities of several endocrine glands. This is very important. Activities of several endocrine glands. In the next part, we will study about that one only, chemical coordination and integration, right? Hypothalamus, which is actually the part of the brain that is also acting as endocrine part. Yes or no? In this way, it is controlling the release of hormones from the pituitary, and pituitary is called as master gland, and that is under the control of again brain, that means hypothalamus. So in this way they are interconnecting all these things, and human behavior is also controlled by the brain. So in this way each and everything will be controlled by the central nervous system. It is also the site for processing of vision, hearing, speech, memory, intelligence, emotions, and thoughts, and all these things are analyzed here and they will be stored here. Clear? Next one, the human brain. Here what I did is, for each and every sentence, as far as possible, I took the diagrams from the internet and I tried to explain it with the help of the diagrams so that you can easily remember this. Okay. Next one, the human brain is well protected by the skull. Already regarding the skull, how many bones, what are the names, everything we studied in the locomotion and movement chapter, right? And inside the skull, the brain is covered with the cranial meninges. These are the layers that are observed. Cranial meninges consisting of outer layer. So now you just here you just observe it. This is the bone, skull bone section, and this is the dura mater. This is the outer layer is called as dura mater, and the middle thin layer is called as arachnoid mater, and the inner one that is actually present very close to brain, that is called as a pia mater. So these are the three names that they have mentioned in the NCERT. Observe carefully. Next one, in the brain they are talking about the three regions: forebrain, midbrain, and hindbrain. These are the three regions that they are talking here. And this is the NCERT diagram. Very clearly they have mentioned each and everything. Observe carefully. Forebrain, cerebrum they have mentioned, thalamus, hypothalamus they have mentioned. In the cerebrum they have mentioned the cerebral hemisphere, and cerebral hemispheres are connected by corpus callosum. So that also they have mentioned. Okay. Next one. Here this is the midbrain they have mentioned. Under the hindbrain total three are there. Pons is present, cerebellum is present, and then medulla oblongata is present, and this medulla oblongata is continuous with spinal cord. And these are the things that are present. And this midbrain portion, whatever is there, now inside this midbrain portion there is a canal that is passing. That canal is called as cerebral aqueduct. This point was also mentioned in NCERT, so remember this. So these are the some parts that they have labeled in the NCERT, so observe carefully. Now first we will talk about the forebrain. So in the forebrain we are talking about the cerebrum, and then thalamus, and below that will be the hypothalamus, thalamus and hypothalamus. Okay, these are the things. So now first we are talking about the cerebrum, and the cerebrum at the center it is having a fissure, so that it is dividing this into two parts, and they are called as cerebral hemisphere. So one is the left hemisphere, other one is the right hemisphere. Okay, this is what they are talking. Next one, both the hemispheres are connected by these nerve tracts, and they are called as the corpus callosum. Okay, this is what they are talking. And next one, they are saying that in the brain there are many folds. This cortex is having, cerebral cortex is having many folds. Okay, and these folds, so when we are talking about the folds, the upper parts are called as, the elevated portions are called as gyri. Okay, plural is gyri, singular is gyrus, and the depressions are called as the sulcus or sulci. So in this way we are giving names, but these names were not mentioned in NCERT. Simply they said that it is having some folds, but these are the names. If possible, try to remember these names. Okay, so this is what they are saying. Next one, they are talking about the white matter and the gray matter. Simply I am talking about the main basic points. Observe carefully. What is this white matter and gray matter? Now you need to think about the myelinated neurons. For the myelinated neuron, please observe this. In the neuron, major length will be occupied by the axon, and the cell bodies are very, very small. And here what exactly happens is, outside is the cortex, yes sir, the cerebral cortex. Okay, and in the cerebral cortex portion, cell bodies and dendrites are present. That means mainly it is the cell bodies. Cell bodies means cytons or cell bodies are present in the cerebral cortex, but the axon portion is present in the inner side. So since axon is myelinated, and who will form the myelin in the central nervous system? It is the oligodendrocytes. Please observe carefully. And this is because of the myelin that is present in the axons, it is getting the white appearance. And in the case of the cell bodies, myelin is not present and they are giving the gray appearance. So that's why with respect to the brain, if you are talking for the brain, at the center you are observing white matter. On the outer sides you are observing gray matter. But opposite is the case for the spinal cord. In the case of spinal cord, at the center, this butterfly like structure, that is the gray matter, but the outside is the white matter, because here the cell bodies are present at the center and this myelinated axons are present on the outer sides. But regarding spinal cord, they have not given information in NCERT, but remember that it is exactly opposite is the case for the spinal cord with respect to brain. Simply remember this basic information. Then they are talking that the cerebral cortex, now it is having different regions like motor areas, sensory areas, and there are some areas they are not either sensory, not motor, but they are actually some interconnection complex network like structures where generally some information processing is taking place, and they are called as association areas. Sensory areas, they are helping in receiving information from various organs, and now this information will be processed mainly in these association areas, and then it will be going to the motor areas. From the motor areas, actually the motor signal will go to the corresponding organ. So in this way, different areas are present within this cerebral cortex and they are associated with various functions. That's what they are saying. And complex functions like interassociation, memory, communication, all these things are associated with association areas. This is what the simple NCERT example they have given, sentence they gave. Try to understand this. Next one they are talking is regarding white matter. Already I told about this. But when they are talking about these things, they used a particular word. So please try to understand this. Fibers. These neurons are the fibers because they are having the long fiber-like structure. Actually, technically, if you are speaking, it is the axon of neuron that is the fiber. But overall neuron also you can call as the fiber. Okay, fibers of the tracts. The term tracts they have used, nerve tracts. They are actually called as nerve tracts. So please remember, this is not just a general word. It is a technical word they used. So I will tell you. So please observe carefully here. Where actually we are using the term called tract, observe carefully here. The group of cytons means cell bodies. Group of cell bodies in the central nervous system, it is called as a nucleus. Plural is nuclei. And in the peripheral nervous system, group of cell bodies. Okay. Sorry. I have changed it. So it is not like this. So group of axons in the central nervous system is called as nucleus. But the group of axons in the central nervous system is nerve tract. Please write these two names here. Please exchange here. Sorry, I made a mistake while typing this. So this you need to write. That means in the central nervous system, group of axons is called as a nerve tract. Please understand this. In the central nervous system, group of cell bodies is a nucleus and the group of axons is called a nerve tract. And use this word here. In the peripheral nervous system, group of cell bodies is called as ganglion or ganglia. Okay, and group of the axons is called as a nerve. Okay, please, there is a small mistake, so please interchange these words. Okay, so this is what the general information. So here they are talking about the tract means it is the in the central nervous system, it is the group of axons. So that's why technically they use these words. So observe carefully. Fibers of the tracts are covered. So tracts means these are the group of axons present in the central nervous system. They are covered with the myelin sheath. Now you can understand this. Okay, and which constitute the inner part of the cerebral hemisphere. For the cerebral hemisphere, inner side this is present. So that's why it is giving opaque white appearance. That's why it is called as white matter. Whereas the group of cytons in the central nervous system is called as nuclei, and that is having the cell bodies that is giving the gray appearance. That's why it is the gray matter. Understood or not? So that's why nerve tracts are having white appearance and the nuclei are having gray appearance. Like this also they can ask. So observe this content carefully. Next one, the cerebrum. The cerebrum is wrapping a structure that is called as the thalamus. So this is the cerebrum, that is entirely it is wrapping this particular structure called as the thalamus. And what is its main function? It is the coordinating center for sensory and motor signals. So many things are passing through this. That's why it is coordinating all this. Next one is the hypothalamus. Hypo means below the thalamus. So this is the thalamus, this is the hypothalamus, present below the thalamus. That's why it is called as hypothalamus. And this is having various controlling centers for the controlling of body temperature, urge for eating and drinking also. And it is also containing several groups of neurosecretory cells, and this information is related to chemical coordination, endocrine system, right? It is having various neurosecretory cells. They are producing various types of hormones, and which secrete hormones, they are called as hypothalamic hormones. And this particular part separately in a different chapter we will talk. Okay, these are the functions of hypothalamus. And here the inner parts of cerebral hemispheres and group of associated deep structures like amygdala and hippocampus etc. form a complex structure called as limbic system. So these are the ones that are forming this limbic system. And what is the role of this limbic system? Along with hypothalamus, it is involved in the regulation of sexual behavior, expression of emotional reactions like excitement, pleasure, rage, and fear also. And even the motivations are also. All these are under the control of this limbic system. And this is the paragraph given in the summary. And here they said that it is concerned with olfaction, autonomic responses, and regulation of sexual behavior, expression of emotional reactions and motivation. This is the another sentence that they have given with respect to limbic system in the summary. So read this particular one also. Here they have added some other sentence, some other words. So please try to remember this one also. Next one is regarding the midbrain. Okay, so here this is the labeling of the midbrain. So this is the middle brain. So this is what the forebrain, midbrain, and then will be the hindbrain. With respect to midbrain, what they are saying, it is located between. So this is the thalamus hypothalamus, between thalamus hypothalamus and pons. Between this midbrain is located. So this is what they are saying. And next what they are saying, a canal called cerebral aqueduct. Cerebral aqueduct, see here, this is the cerebral aqueduct that is passing here. Actually this concept is related to ventricles of the brain. In the brain, there are two lateral ventricles, each one is present in each hemisphere, and then they are connected to the third ventricle. And now this third ventricle is passing through a small canal. This canal is called as cerebral aqueduct. And now this is opening into the fourth canal, fourth ventricle. Now this fourth ventricle is continuous with central canal of spinal cord. In this way, so the ventricles concept and with respect to cerebrospinal fluid formation and the movement, there is a different concept, but this much was not explained in NCERT. Simply they said that this midbrain is surrounding this central cerebral aqueduct, or reverse, cerebral aqueduct is present at the center of this midbrain. This is what they are saying. At least this information you please remember. Clear? Next one, the dorsal portion of the midbrain consists of mainly 4 round swellings. Okay, and these are actually called as, upper 2 are called as superior colliculi and the bottom 2 are called as inferior colliculi, and these colliculi names are also not given, and all these 4 swellings are commonly called as corpora quadri. Quadra means 4, quadrigemina. Okay, and actually upper 2 are involved in the responses of the body with respect to the vision, and the lower 2 are involved in the response of the body with respect to the hearing and all. So in this way they are having different roles, but not mentioned in NCERT. At least NCERT points you please remember. The midbrain receives and integrates visual. Visual means vision related, tactile means touching related, and auditory means sound related inputs. So with respect to these things, so it will try to give some information so that our body will respond to these particular signals. This is what they are saying. Finally come to the hindbrain. In the case of hindbrain, total 3 parts are there. What are these? Pons, also called as pons varoli, and medulla oblongata, and then cerebellum. These are the 3 things combinedly they are forming the hindbrain. Okay, so these are the 3 things they are forming the hindbrain. And pons consists of, this is the pons or pons varoli, we can say it as. Pons consists of fiber tracts that interconnect different regions of the brain. So since many of these are passing through the pons only, so it is interconnecting the neurons that are related to the different, going to the different regions of the brain. And cerebellum original meaning is, it is like a small cerebrum. Like the cerebrum, this is also highly convoluted, highly folded structure. Okay, it is inside also, it is having a tree like structure and all, so that it can increase the space, so that it can accommodate more and more neurons, and this is also very important for balance in many things, but much information was not given in NCERT. Cerebellum has very convoluted means a folded surface in order to provide additional space for many more neurons, so that many more neurons can be accommodated here. And the cerebellum integrates information received from the semicircular canals of the ear. Semicircular canals of the ear are related to the body balance. So cerebellum is related to the body balance, and the auditory system, even from the hearing, also related to this particular part. This is what they are saying information. So observe very carefully. Next one. So here finally they are talking that regarding the medulla. It is also called as medulla oblongata, and actually it is continuous with the spinal cord. The medulla of the brain is connected to the spinal cord. The medulla contains centers which control respiration, cardiovascular reflexes, gastric secretions. So in that breathing and exchange of gases, and even in the case of the body fluid circulation and the regulation, we studied about all these things, right? So observe this particular information. Next one. Three major regions. What are the three major regions? It is the midbrain, and then it is the pons, and then it is the medulla. Midbrain, pons and medulla. These three combinedly they are forming the brain stem, because this is like a stem that is holding the entire brain. So that's why midbrain, pons varoli, and then medulla oblongata. These three. So three major regions that make up the brain stem. They are the midbrain, pons and medulla oblongata. So they are actually forming the brainstem. Brainstem forms the connection between the brain and the spinal cord. So in this way, these two are connected, and also it is holding this particular structure. So this is what the basic, general, simple information they have given in NCERT. My suggestion is, please focus on each and every word and sentence of NCERT. That much is enough. If you start going out of NCERT, there is no limit for this. Unnecessarily, you will end up with many more problems. That's it. So my suggestion is, fix and stick yourself to NCERT only. That's it. With this particular chapter is completed. Before concluding, once again I'm telling you that, so these revision lectures are available both on Live Prep app and also on YouTube channel, but test series can be given only through the app. With this part is completed. So in the next lecture we will meet with a different new chapter revision. Thank you.